పంపిస్తున్నా.
We will join the faculty only.
Amma, somebody just send a link, no? A regular college ka link.
Yes ma'am, evariki పంపించేసేయాలి ma'am?
నాకు పంపియండి. ZCO మేడం కి నాకు పంపండి ఒకసారి.
పంపిస్తాను ma'am.
నాకు పంపిస్తే అది 861 కి పంపిమా.
రెండిటికి పంపిస్తాను ma'am. పంపించను ma'am.
అమ్మా రజిత వీళ్ళు జాయిన్ అయ్యారా?
లేదు మ్యామ్, ఇంకా జాయిన్ కాలేదు.
మ్యామ్ నేను సుమతి ఉన్నాం మ్యామ్, స్వరూప మ్యామ్ ఉన్నాము, రజిత మ్యామ్ ఒకరు లేరు.
అమ్మ కో-హోస్ట్లు కొద్దిగా చూసుకోండి, అడ్మిట్ అవుతున్నారు.
Sorry for the inconvenience.
Apologies.
మా నిరూప
మేడం,
ఇప్పుడు చూసి మనోళ్ళనే, వేరే వాళ్ళు ఎవరిని యాడ్ చేయకండి. మన సార్ వాళ్ళని చూడండి. అశోక్ వాళ్ళకి పంపించాను, విష్ణు కూడా పంపించినాను నేను చూసాను. అందుకే విష్ణుకి అప్పచెప్పమన్నాను. ఇది వెళ్ళిందా?
ఇప్పుడు మా ఆఫీస్ లో కూడా మేనేజర్లు, అలాగని ఇంకా పెద్ద మేనేజర్లు వీళ్ళందరూ బ్రాహ్మిన్స్. మామూలుగా మీరు ఒక స్టేట్ నుండి వచ్చారు, మీకు మేనేజర్ పోస్ట్ దక్కాలంటే మీరు 25 ఇయర్స్ రాదు. అట్లుంటుంది.
రజిత మ్యామ్ జాయిన్ అవుతున్నారు మ్యామ్. ప్రవీణ్ సార్ కూడా జాయిన్ అవుతున్నారు.
మామిడాలనా ఆర్ఎస్ ప్రవీణ్ కుమార్ సార్?
మామిడాల సార్ జాయిన్ అయ్యారు.
Good morning, sir.
Sir, good morning, sir.
Very good morning, sir.
There was a lot of inconvenience, sir. I'm really sorry. The thing was, there are 1,300 participants. 1,300 people came at once. And because of that, waiting list, we couldn't see who is coming and who's not. There were 1,300 registration at a stretch. That was a problem mainly.
Okay.
So we had to change the ID.
Whether today's resource persons or the keynote speakers have joined the session?
No, sir, they haven't joined as yet. And we have sent the new ID and password. It will be done, sir.
So please send apologies to them because they are the very senior most scientists. I looked at their profile in the LinkedIn. So please send our apologies that there are some technical glitches and to join in the other link. Okay.
Okay, sir. Okay.
Yeah.
మ్యామ్ అశోక్ సార్ జాయిన్ అవుతున్నారు మ్యామ్.
Right.
హలో మ్యామ్. మ్యామ్ ఇది కోహోస్ట్ ఐ మీన్ స్క్రీన్ షేరింగ్ ఇవ్వాలి మ్యామ్.
Yeah, if Rajita has joined, can she play the PPT for some time so that it doesn't?
Ready, ma'am?
Yeah, ma'am. You please play the PPT.
Yes, ma'am. Perfect. Thank you, ma'am. Thank you for the opportunity. And respected dignitaries on the other end, I'm graced with your presence, and it's a wonderful opportunity for me to be presenting this PPT. Welcome you all once again. Welcome to this international webinar. This is Raji Reddy, lecturer, Department of English. Now, welcome to Social Welfare Family and International Webinar once again to all of you. Coming to the contents of the presentation today, the first thing is about social welfare. The next point is the man behind the success story, the 10 commandments, vision and mission of our great institutions, achievements about TSWRDCs, and finally about Mahendrahills. This is what our tour will be in PPT. Now, the first one is about the social welfare institutions. Our social welfare institutions was established in the year 1984 when the two Telugu states were united under the leadership of N.T. Rao Garu. At present, there are 268 institutions, including schools, junior colleges, and degree institutions. It is inspired by the man, the real hero behind this vision and mission to enlighten every caste, beyond caste and creed, enlighten every man with blood and muscle. That is Dr. B.R. Ambedkar Garu, the father of Constitution. And next, inspired by Jyotirao Phule and Savitribai Phule, who made the girls to study for the first time in their life and run the schools for them. And also revolutionized by our bureaucrat S.R. Sankaran Garu, who had brought the benefit for the underprivileged people and students to get education without any partiality. And finally, I would like to introduce our hero of this entire story, that is Dr. R.S. Praveen Kumar Garu, who is currently heading our institutions, TSWRIES and TTWRIES. Now, the man behind the success story of Gurukulas, Dr. R.S. Praveen Kumar, IPS Garu, Secretary, Telangana Residential Education Society, had been the 1995 IAS batch, and he has been the constant inspiration for our lakhs of students around the state and around the globe, I can say. He was graduated from Harvard University in 2011. Dr. RSP aspires to change the facet of underprivileged students, and he wants one thing, that is unity in education and equality in minds of people. So how to fight poverty? The poverty can be fought with the sword of education. Now I can show you the two great people on the screen who are the real reason for TSWRDCs and higher education department, headed by Dr. Praveen Mahendrahills, TSWRDC, Joint Secretary, entitled that this mission is taken with some tools. The tool you can say as 10 commandments. That is, I am not inferior to anyone. I shall be leader wherever I am. I shall be I should love what I love. I should do what I love. And such inspiring words are said by every student at the beginning of the assembly by placing their hand on the heart. And this, in an interview, Dr. RSP clearly said that gone are the days of calling SCs Chandala, Hachin, Dalit, Badugu, Nimma, etc. And Sweros is our new tag name. Sweros is our new potential, helps us to discover our new potential, and says that the Sweroism should provided for the benefit of thousands. And here, the vision is to build outstanding government educational institutions, provide high-quality, holistic, value-based education, to promote wide range of social extracurricular activities, and then taste varies to be the 21st century educational revolution. That is the dream and vision and mission of our great people behind. Now, types of institutions. You can look at the chart, my dear friends, or that what are the organizations that Taste Varies run, and what are the special schools like School of Fine Arts, Filmmaking, Science School, and Social Welfare Forces Preparatory Schools, and Centers for Excellence, and whatnot. We have been doing more extracurricular activities, which makes you learn. Now, coming to the achievements, we all believe that sky is the limit. In the first picture, when you look at, you can see that the laudable event done by our students of TSWRDCW, where 15 students had launched the satellite and on Tata Institute of Fundamental Research, which is on cosmic radiation, temperature, and ozone concentration. Not just that, we have taken our children have taken number of prizes and appreciation from governors, and many other eminent people around the globe. That is what social welfare is. Now coming to the learning methodologies, you can see here the pictures that how learning is done. It is learning by doing, my dear friends. It is never a rote method or a lecture method or a textbook method. It is always flipped classes. It is always we aim to our children to study in internships in international universities like CCIP, AISCC, etc. And we also have our secretaries here and other eminent people. And when coming to the next slide, we can observe that Purna Malawat and Anand in a pic with our Prime Minister Garu, which means that a girl from tribal Tanda has aimed to climb Mount Everest and now aiming to become an IAS officer. That is the spirit that which shows that think big and aim high and never fear the unknown. The true commandments are being realized. They are not just been said. They are realized in our real life situations by our students, by our faculty and family of social welfare. And now we can see the Pride event that recently been achieved by our social welfare. It is a feather in a cap, that is VLC, Village Learning Center. We have to stand and salute all these girls beyond risking their lives. Even in Corona, they have started tuitions. They have started mentoring students in VLC. Every village has got a VLC and monitored by Swero Network, and teachers are visiting it. Our beloved Secretary Sir and Mamdala Sir and other dignitaries have visited it. And there is a lot of change and transformation in educational system just because of this VLCs. Hats off VLCs. And then next slide, we can see about TSWRDCs. TSWRDCs is a story, the story of pain. When a pregnant girl encountered Dr. RSP saying that why can't girls study further? Why should we be a prey to marriage? And she spoke in proper English. This made him to think that why can't inter girls get a platform to study and fly high? And hence he decided no other women, no other child of 18 will go into marriage unnecessarily. And that is why social welfare degree colleges are established. And the ambition is think big and aim high. I repeat, think big and aim high. And with a view of augment facilities for the girls to cater higher educational needs, Telangana's government, in collaboration with social welfare, has established this degree colleges, and we have 30 degree colleges around so far and aiming to procure education further. Now about Mahendrahills. Mahendrahills is a specialized science college established in 2017 as a special branch of education only in life sciences. It is a residential model and with a proper ambience located at Gatkesar, and the courses offered are 7 here. For now, from this year, we have 6 courses, and they are BSc, BZC, BCCS, MBZC, NDZC, BGC, BTBC, and BCZC, with high qualified teachers and entirely curious students, and there is an obvious symbiotic relationship of proper scientific thinking. Now you can see the atmosphere of how our college looks like. This is the wonderful building and the amenities and ambience that our organizers have provided. Here is a small intro about ourselves. And
Raji Reddy, ICT.
And I thank you one and all, and this is where I want to end. And over to Principal Ma'am for giving me this opportunity, and all the dignitaries over. Thank you. Thank you so much.
Good morning, all of you. And firstly, I just apologize all of you for the inconvenience caused. There was a huge response from the participants because of the high profile of the speakers and all. A maximum of 1,300 of them have joined at a time, and that caused a greater inconvenience to all of us. So I first of all, I deep from my heart, I apologize the speakers, Dr. Mahender, Dewal, and all JS sir, and all the dignitaries over here for delaying the process and all. And I welcome all of you, sir. And I first of all thank all of you for joining this session. And in spite of all these things and things you have, with a greater heart, you have joined the webinar to make it a great success. Thank you so much, sir. I take this occasion to invite our Secretary, sir, Dr. Praveen Kumar, sir, and Dr. Joint Secretary Praveen Mamidala, sir, and the speaker of today, Dr. Mahender Dewal, and Raji Reddy Garu. Also, I would also like to welcome Dr. Ashok, Professor Usmania University, who had taken pains for conducting this meeting and all. And one more thing, I am the student of Dr. Ashok, sir, and for this, I'm really thankful to Ashok, sir, also. And I welcome each and everyone. And now I hand over the session to Zonal Coordinator, ma'am, for the further for presiding over the meeting. Thank you once again. And once again, I apologize all of you for the inconvenience caused. Thank you, sir. Thank you so much.
Thank you, Nirupa.
Zonal Coordinator, ma'am.
Thank you, Dr. Nirupa. Good morning, everyone. First of all, I want to start by apologizing for the inconvenience created now, and somehow we could sort it out. And, we are immensely sorry for the same. I am Dr. Rajini, Zonal Coordinator for South Zone, TSWRDCW, Telangana, India. I am immensely happy to see so many overwhelming people coming and joining in this for this international webinar, which has created a little bit of inconvenience also. Even then, I'm very happy to see so many people wishing to join. And our thanks to all the registered care delegates for supporting this webinar on role of chemistry in combating COVID-19. On behalf of Telangana State Social Welfare Residential Degree Colleges, I extend a warm welcome to our Honorable Chief Guest, Dr. R.S. Praveen Kumar Sir, IPS, and Secretary, TSWRDCW and TTWRDCW. Thank you, sir, for accepting our invitation in spite of your busy schedule. I also welcome the distinguished special guest, Dr. Praveen Mamidala, Joint Secretary, TSWRDCW. Thank you, sir, for accepting our invitation and encouraging us to conduct this webinar. I also welcome our OSD, Bhanuprasad Garu. My heartfelt welcome to our resource persons, Dr. Mahender Dewal Garu from MIT, US, and Dr. D. Raji Reddy Garu from IICT for accepting our request to be our resource persons for today's international webinar, Role of Chemistry in Combating COVID-19, in spite of their busy and hectic schedule in research field. Thank you once again, sir. You all very well know that the country and the world is struggling against a dreadful pandemic caused by coronavirus 2. And you also very well know that it was first discovered or identified in December 2019 in Wuhan city, China, and it has now resulted in an ongoing pandemic COVID-19. As per WHO today, about 30 million corona cases have been reported across 200 countries, resulting in more than 7 lakh deaths. India being the third highest in death rate in spite of implementing lockdown in advance. Coming to Telangana, more than 1 lakh corona cases reported, and day by day cases are being increased even in rural areas. So many people are eagerly waiting for specific new drug and a vaccine for the effective treatment of COVID-19. Now, chemists play an important role here in discovering the drug or the vaccine, and worldwide, a large number of chemists are working on the discovery of the drug or the vaccine. Till today, there is no specific drug or any vaccine released, which is being potent and effective in reducing the severity. Research is going on, and in many countries, trials are in either in second phase or in third phase. Recently, Russia also has released a vaccine of the second phase, and many countries are not accepting it. Therefore, I feel our webinar will give a clear picture to the participants about the research going on in the world today and where we stand now. Thank you very much. Now, I request our JS Garu, respected sir, I request you to give an introductory speech. Thank you, sir.
Very good morning to one and all, my dear colleagues. Thanks to Mrs. Rajita, lecturer in English, for excellent presentation on vision and mission of our society. Because of powerful speakers today, Dr. Raji Reddy Garu and Dr. Mahender, Dewal, for both of them, their participation, the Zoom session was house full. Around 1,300 students at one go, they joined, and they got lots of technical issues. I was in queue for more than 40 minutes, my dear friends. Finally, I got a chance to join you all in this wonderful session on role of chemistry in combating COVID-19, and a very excellent webinar and aptly you are organizing. Today is a special day for me, my dear friends, because both speakers are with a chemistry background and both are from Telangana and had a great global experience and did a lots of very, very good research. I also welcome a senior most professor in this group, Professor D. Ashok Garu, Usmania University professor, who was a former registrar of Telangana University. Very good morning, sir. And I appreciate the Journal Coordinator, Dr. Rajini ma'am, for her commendable efforts in organizing several national and international webinars since last 3 to 4 months. Also, Dr. Banu Prasad, OSD, who is a chemist, Dr. Nirupa, principal, who is a chemist. All people, including our JETSU, all are the chemistry background, and that's where we are having a wonderful session on this COVID-19 pandemic. I especially thank the speakers today because the moment when Dr. Rajini told me that they are inviting these speakers, when I Google their profile, it's awesome. They have got a huge research. When I look into this Chada Raji Reddy Garu, a senior principal scientist from IICT, PhD and postdoc from abroad with more than 100 of research papers and 7 international patents, and active in green chemistry and active API, and lots of research he has done. Thank you, sir. Thank you very much for joining us and empowering our students. And I also see this Mahender B. Dewal, doctor, is a founding member, head of research and development at Expansion Technologies, Cambridge, and who's a research fellow at MIT, one of the premier institute, Massachusetts Institute of Technology is one of the premier institute, and who also have a postdoc experience at Wayne University, Detroit, who is a specialist in the interdisciplinary like biochemistry, molecular and cell biology, chemical biology, and enzymology, medicinal chemistry, molecular modeling, organic chemistry, and more than that, huge, very good publications in high impact factor journal, and 4 US patents. When I looked into profile, I was very happy that he also taught UG students in Nizamabad, so where I worked for some time in Nizamabad in Telangana University. And thank you very much, sir, for all of you for joining us for this today wonderful session. As a biology teacher, I must thank coronavirus. Really, I should thank the great coronavirus, because now at least government of India has opened its eyes and came to know the importance of science, particularly in the research institutions, not only in the research institutions per se, but also in the universities. Because since last 10 years, we have Professor Raji Reddy Garu and also we have Professor Ashok in the today's meeting. There is no much funding either from DST, DBT, or UGC, or AACT, whatever the SERB, whatever the funding agencies you take, they have ignored the science, and now they have done very bad. So it's all because of science. It's all because of research. We can do wonders. We can do lots of innovations. And top on this, we need lots of interdisciplinary research, which is really missing in our, particularly in the so-called developing nation, India, my dear friends. My dear friends, we have more than, I can say, dangerous health issues than corona. We are unnecessarily worrying about corona since last 6 months. It is just like we are feeling it's a huge pandemic, but of course it's a pandemic. But other than this, we have huge diseases like diabetes. Diabetes is killing lots of people. Cancer killing lots of people. Tuberculosis. Obesity has recently entered into our nation 10 years back. Now most of Indians are suffering with obesity. If we have a very good interdisciplinary research like chemistry combined with life sciences, with say physical sciences and modeling, we can easily wipe out this dangerous coronavirus. Only thing is we all need to do is to think differently and chalk out what are the areas or how to inhibit the replication of coronaviruses when enters into the human cells or without making much damage to the human system, and utilize all the computational tools and design a very good drug to inhibit this. Lots of research is going on, my dear friends. I think there is no need for me to say more on corona because we have wonderful speakers today. My only request to all these speakers is, sir, we have a wonderful ecosystem now under leadership of our Secretary, sir, Dr. R.S. Praveen Kumar. Though he's IPS cop, he is for me, I feel that he's 100 times better than the top-notch vice chancellors across the country. He knows what to do, how to repair, and how to make a very good educational reforms. And in the recent past, we made a very good interaction session with the lecturers, and we are trying to establish a research council. I request you in this session, you should be part of the research council, so that we want to encourage our degree lecturers to establish more of research-based experience teaching. More of, just like in ICERS, just like in NICER, just like in some, if you take a private institutes like Ashoka University, where an undergrad student will start doing research. He will try to publish a paper at the end of his undergraduation. So that type of ecosystem we are going to establish. So we need your help to join our research council. I will get back to you again. But once again, I really thank you, and special namastes to Dr. Raji Reddy Garu and Mahender Dewal Garu for joining us this session, and special thanks to Professor D. Ashok sir also, because he was my teacher. So thank you very much, and over to Nirupa. Thank you, sir.
Thank you, sir. Thank you, sir. Thank you for your valuable speech and all, and enlightening everyone into this thing. And also thank you for appreciating our efforts also. Next, I would like to hand over the session to Zonal Coordinator Madam for presiding over the webinar.
Before the zonal coordinators addressing a small information, my dear friends, Secretary sir has tried his best to join this meeting from 8:50 to 9:10 because he always follows time. We have a practice of Sparrow Standard Time, but because of this technical issues, he has not joined. Now he has entered into some other meeting, so he just conveyed me that to wish you best wishes to the organizers, and also best wishes to the JETCO, and special thanks to all the speakers today. So he may not join us. Okay, thank you very much.
Thank you, sir. Really, we missed our Secretary sir's speech, and in his absence, I want to say a few words about him. He being IPS officer, our Secretary sir, that is R.S. Praveen Garu, IPS, he has chosen to education because he firmly believes that only education leads to equality and fraternity, and gives justice. The brain behind this, and he's the main person and the brain behind the success story of the Everest conquerors. That is Malavat Purna and Sandanapalli Anand Kumar. 13 years age of tribal student from Nizamabad, Telangana, created history on 25th May 2014. And she, that is the Purna, became the youngest girl in the world to scale the Mount Everest. and we are proud to say that she is our student. And he has done so many things. Our secretary sir has transformed social welfare education system into the world-class learning centers. Now he has introduced VLCs also in this crisis. He has his thoughts are the main in creating village learning centers also. And we are thankful to our beloved secretary sir and to have him as our secretary. Thank you, sir. Thank you once again. And now may I call upon Dr. Pavani, lecturer in zoology, to introduce the first session resource person, Dr. Mahender Dewal garu. Dr. Pavani?
Yes, ma'am. Thank you, ma'am.
Yes, go on.
Yeah. Hello, everyone. Very good morning. I'm Dr. Pavani, faculty in zoology from TSWRDC Mahendrahills. I'm very excited to be here in this program with all these eminent personalities. It is in fact, my privilege to introduce the first speaker for today's event, Dr. Mahender B. Dewal. I was truly amazed looking into his journey so far. Even I looked up at LinkedIn and I was really amazed, sir, looking at your journey. He started his career as an undergraduate student at our very own Osmania University. And today he's with us as head R&D, principal scientist and founding member of Expansion Technologies, Cambridge, the United States. And I would like to take this opportunity to share a few of his career milestones for our audience here. After obtaining his MSc in Organic Chemistry from Osmania University, Dr. Mahender worked as a junior lecturer in chemistry in Kakatiya College, Nizamabad. Later, he joined as a research fellow at Indian Institute of Chemical Technology, where he worked on the synthesis of D-serine from N-acetyl D-glucose using microbial enzymes. In 2003, he joined as teaching assistant in the Department of Chemistry at University of Kent, Ohio. Later, he moved to the prestigious University of South Carolina, Columbia, from where he also obtained his PhD. He associated with the Department of Pharmaceutical Sciences, Wayne State University, the United States, for his postdoctoral work, where he worked on structure-based design and synthesis of small molecule protein-protein interaction inhibitors for the treatment of human cytomegalovirus infections. His association with a world-class institute like MIT reflects his caliber and credential in his area of work. In 2015, he founded Expansion Technologies, a company that is revolutionizing the early detection of diseases through novel super resolution imaging methods. From 2016 to till date, he has been acting as head for the R&D department of the same company, Expansion Technologies. I hope I was able to do justice for the speaker with my little introduction and looking forward for a wonderful and an interactive session, sir. Over to you, sir. Thank you.
Thank you. Thank you so much for a great introduction, Dr. Pavani. Thank you so much. Though it is a one day belated, happy Teachers' Day to all the teachers and lecturers, particularly my guru, Professor D. Ashok Garu, who taught me organic chemistry during my undergrad. Can everyone hear me well?
Yes, sir. Yes.
Yes, sir.
Yes, we can
Okay,
all
good.
hear you.
Thank you.
Yes, sir.
Excuse me. Good, thanks. So, good morning to everyone. First of all, I would like to thank Telangana Social Welfare Residential Education Institution Society, each and every person, including Professor D. Ashok Garu, for inviting me to give this talk today. It gives me an immense pleasure to speak to all the social welfare students today, because I'm none other than one among you. My entire education was done in all government schools and government colleges in Telangana state, that too in Telugu medium, until I get to OU campus for my master's. Then I came to US for my PhD on a fellowship as well. So do not hesitate to contact me or reach out to me to get my time in future to address your students or you. I'm happy to do so. Also, I would like to thank our chief guest, Dr. R.S. Pravin Garu. Unfortunately, he was not able to make it, but for his valuable guidance to the society. And Dr. Pravin Mahendrahills Garu for as a joint secretary for his valuable time to the society. Thank you. Thank you so much for your gracing this event with your presence. And also, this event is unique for me. What you guys did is not just invited me to give a talk, but also you made me to meet my old friend, Dr. Raji Reddy, a wonderful scientist who was my classmate at OU campus during my MSc. So after 25 years, we said hi to each other. So this is a unique event for me as well. Without further delay, let me share my presentation and then we'll get going. Can everyone see my slides? Fine?
Yes, sir.
Just someone say yes or no.
Yes, sir.
All right.
Very clear.
Awesome. Cool. All right. So the title of my talk today is COVID-19, a pandemic of coronavirus, progress in therapeutic and vaccine development. First half of the talk, I'm going to talk about the evolution of coronavirus and their infection mechanism. And also I'm going to discuss about the immune system components, how they fight with the virus and other infections. In the second half, I will be mostly focusing on therapeutic and vaccine development methods, approaches, and current progress in their development. COVID-19, a 5-letter, 2-number word, has been shaking the world from past 18 months. So what is this COVID-19? COVID-19 is an infectious disease caused by the most recently discovered novel coronavirus. And in mild cases, it starts with most common symptoms such as fever, dry cough, sore throat, diarrhea, headache, nausea, and so on. But since March onwards, the patients infected with coronavirus, novel coronavirus, also complained about 2 additional symptoms, which is losing the smell and taste. Therefore, most of the countries from March onwards included these 2 symptoms also as a coronavirus identification symptoms, infection symptoms. In severe cases, it causes pneumonia, severe acute respiratory syndrome, SARS. That's why this virus is also called SARS coronavirus, which causes organ failure such as kidney and lung, ultimately leading to the death. When I said novel coronavirus, why this coronavirus is called novel coronavirus? Is there any other coronaviruses already existing in the world? Let's take a look into the history a little bit. The human coronavirus were first described in 1960s. Here, I have listed 4 different serotypes of coronaviruses that were 2 among these 4 were described in 1960s, 2 others were later in late 1990s. All these coronaviruses, 4 these serotypes, human coronaviruses, are originated either from bats or from rodents, but they are transmitted to the human via an intermediate host such as camel or cow. These 4 initial serotypes of coronaviruses does cause only mild symptoms when they infected the humans. That's why they are not considered to be highly pathogenic. They only mostly focused or mostly infected the immunocompromised patients. However, in 2002, SARS coronavirus 1, severe acute respiratory syndrome coronavirus 1 broke out in Fushan, China. And this was spread to only 30 countries in 5 continents. That's why World Health Organization announced this as an epidemic, not the pandemic, because it was limited to only a certain number of countries. And the source of this coronavirus is also bat, but it is transmitted to the humans via an intermediate host including the civet cats or raccoon dogs. And this coronavirus 1 also causes mild to severe symptoms, including a death to the patients. Approximately 8,000 people, excuse me, from these 30 countries were infected by this SARS coronavirus 1, having 800 of them were declared dead. Also, in 2012, another coronavirus, Middle East Respiratory Syndrome coronavirus, MERS coronavirus, broke out in Saudi Arabia. And this one also announced by World Health Organization as an epidemic, not the pandemic, because it was spread into about 27 countries. Again, the origin for this MERS coronavirus is also bats, but the intermediate host is different from the other coronavirus, which is a dromedary, also called Somali camel. And the infections from MERS coronavirus is also very dangerous. Mild to severe symptoms it can cause into the infected people. Approximately 2,000 people from these 27 countries were infected by MERS coronavirus, having 700 of them declared dead, approximately with 35% of lethality rate. As most of you already know that, SARS coronavirus 2, the novel coronavirus that I mentioned in my previous slides, broke out in 2019 December in Wuhan, China. Pretty soon, it spread out to entire world and became a global pandemic announced by World Health Organization by March 2020. Again, this coronavirus also originated from bats, but the intermediate host is different, which is a pangolin compared to the other coronaviruses. As of today, approximately 26.6 million people from all over the world were infected by this coronavirus, having 865,000 million people were declared dead. And this coronavirus is also can cause mild to severe symptoms. The disease caused by this coronavirus is called COVID-19. As so far, I have displayed 7 different types of serotypes of coronaviruses. Among those 7, last 3, SARS coronavirus 1, MERS coronavirus, and SARS coronavirus 2 are more dangerous than any other coronaviruses among these 7 serotypes. So, what is the differences and similarities between these 3 dangerous coronaviruses? Let's take a look into that as well. So, one of the most similarities, can somebody mute the guys, whoever is making some noise? It's very disturbing. You can mute the people probably, or they can mute themselves. Thank you. So the most common similarity is 90 to 95% of the genome of these 3 coronaviruses is similar to the coronavirus that is found in bats. That's why it is originated from the bat coronaviruses. And also, the SARS coronavirus 2, the genome of SARS coronavirus 2 is 80% of its genome is matches or similar to the SARS coronavirus 1. They are like brothers, siblings in the same family. And only 50% of the SARS coronavirus 2 genome matches with MERS coronavirus. They're like cousins. They're not in the same family, but they're cousins. And there is also another interesting or important similarity between SARS-1 and SARS-2 is the cell entry receptor. Angiotensin converting enzyme 2, they use this ACE2 receptor to enter into the human cell to infect the humans. But the MERS coronavirus uses a different cell entry receptor, which is DPP4, dipeptidyl peptidase 4. That's why this is different from the 2 other SARS coronaviruses, SARS coronavirus 1 and SARS coronavirus 2. However, among these 3 highly infectious coronaviruses, SARS-2 is even much more rapidly spreading compared to the MERS and SARS-1. If you have looked at the numbers that I mentioned in my previous slides, what could be the reason? Let's take a look at that. So why this is rapidly transmitting compared to the other 2 coronaviruses? Excuse me. The primary spread is person to person for these coronaviruses by coughing, sneezing, or speaking. The respiratory droplets that come out of the coughing and sneezes are much more infectious that can infect the people. That's one of the primary reasons. The secondary spread is contaminated objects or surfaces. If you look at the table here, I have listed that the stability of the SARS coronavirus 2 on various different surfaces at various different temperatures. Let's take a look at that. At 4 degrees centigrade, that means in the fridge, this SARS coronavirus 2 is stable up to 2 weeks. At 37 degrees centigrade, that means approximately a body temperature, not inside the body, outside the body, it is stable up to 2 days. But at 70 degrees centigrade, approximately close to the boiled water, it can be deactivated or cleared out within 5 minutes. But at room temperature, 22 degrees centigrade, which is called normal room temperature, the stability of SARS coronavirus 2 is much more different from, it depends on the surface. If you look at the regular paper, tissue paper, it survives up to 3 hours at room temperature. However, the currency note and a cardboard, these two are also made of paper, but the stability is much more different. Up to 2 to 4 days, it's stable to infect the people. And if you look at the glass, plastic, can stable up to 7, stainless steel, 7 days, cloth, wood, up to 2 days. So this is also causing the rapid spread of this coronavirus. It's stability on various different surfaces at various different temperatures. And long incubation period is also one of the important factor. Up to 2 weeks, a person who is infected may not show any symptoms, but he can rapidly spread the virus infection to others. Recently, it has been found that the mutations also causing a rapid spread of this coronavirus, particularly aspartic acid, D, at 614th position of a spike protein is mutated into glycine amino acid. Here, this is a spike protein, a cryo-electron microscopy structure of the spike protein, which has a 2 subunits, S1 subunit and S2 subunit. And this mutation is happening at the junction of 2 subunits where the 2 subunits join. So this is the aspartic acid, 614 amino acid, which is mutated into glycine, and that is rapidly spreading the virus. When experimentally it was tested, the 2 viruses are made in the lab. The one with the wild type aspartic acid mutation, and the second one with the glycine mutation. This is the wild type, and this is the mutation. Among these two, when the human cells were infected with glycine mutation, it was much more rapidly infected compared to the within minutes to hours compared to the wild type mutation. And also, if you look at the data from the real-time virus sequencing data collected from the patients who are infected with the coronaviruses, in January and February, there is no mutations happening. Only wild type virus was found in the people who were infected with. But the mutation started taking place in March, about 26%. April went to 66, 65%. May, 70%. Now it's almost 90% of the virus that is infecting people is the mutated virus, which is the glycine 640 mutation. So let's take a look at what is the coronavirus. What is this? Much more details in the coronavirus. So once Peter Medawar, he was an immunologist, a Nobel laureate, defined virus as simply a piece of bad news wrapped up in proteins. Exactly. Like living cells, coronaviruses or any other viruses contain genome. It could be a DNA or it could be a RNA. In case of coronavirus, it is a large positive sense RNA, which is very well securely protected by a nucleocapsid protein. And unlike living cells, the viruses does not divide into multiple of them. It requires a host to replicate itself. Like bacteria or human cells, they divide in 1 to 2, 2 to 4, 2 to 4 to 16 or like that. But viruses does not divide like that. It requires a host to replicate its RNA and become infectious viruses. And if you look at the structural proteins on here, it has very uniquely defined structure protein, which is a spike protein. And that's why these spikes are looks like a crown. That's why this one, the virus is called coronavirus. And it also has envelope protein, membrane protein, which gives a very well organized structure to the coronavirus. This virus belongs to the zoonotic family. Excuse me. That means this virus exists in the animals, but it can infect animals to animals, animals to humans, and humans to humans. The transmission can be happening. So when I say the genome is very well securely wrapped in the nucleocapsid protein, let's take a look at much more closer look into the genome of what is in there. So as I mentioned, it has a large positive sense RNA with 29,000 nucleotides encoding approximately 29 proteins. And this genome is a 2D map here, which can be divided into 2 parts. First one has 2 open reading frames, which encodes 16 non-structural proteins that has a very crucial role in function of this virus. And the second part contains 4 structural proteins and 9 accessory proteins. Among the 16 non-structural proteins, as of now, these two proteases and another enzyme which is called RNA-dependent RNA polymerase, RDRPR, which is also called a replicase, has been very well characterized, but rest of the others are not well characterized. And also a couple of other NSP1, NSP8, NSP7 also and are characterized well. So among these proteins, non-structural proteins, proteases help the virus to cut into small proteins so that they can function functionally very well active when it is translated from one long polypeptide chain. The most important enzyme is RNA-dependent RNA polymerase replicase, which transcribes the virus RNA into a negative strand without any errors, which plays a very crucial role in virus replication. It takes the help from NSP7, NSP8 non-functional proteins. NSP1 has a very crucial role in protecting the virus from immune system that I'm going to explain you in my upcoming slides. The accessory proteins. We do not know the functions are not clearly defined yet. There's 9 accessory proteins, but the structural proteins are also very well defined. They have a very crucial role in the viral infection. Nuclear capsid protein protects the genome of the virus. Spike protein is very important, very crucial in for virus to enter into the human cells by attaching to the ACE2 receptor enzyme. Envelope protein, membrane protein provides the structure. So let's take a look at the cell entry mechanism. And here, this is the cell membrane. As I mentioned in my previous slides, coronavirus uses its spike protein by binding to the ACE2 receptor down here to enter into the human cell. Once the spike protein bound to the ACE2 receptor, where the endosome will be formed. Once the endosome forms, the virus goes through certain mechanisms, certain changes in the spike protein, and it fuses into the cell membrane and releases the RNA into the cytoplasm of the human cell. Entire this process is called endocytosis. Here is the cartoon of the interaction between a spike protein and ACE2 receptor on the human cell, spike protein on the virus. The spike protein has 2 subunits, as I mentioned in my previous slide, S2 subunit and S1 subunit. Among the S1 subunit, this particular region, which is called receptor binding domain, plays a very, very important role in binding strongly to the ACE2 receptor to enter into the human cell. Once it releases the RNA in the human cytoplasm, immediately the cellular enzyme starts translating the first half of the genome of virus genome, which has the open reading frames, into the functional non-structural proteins like proteases and replicase. As soon as the replicase is made in the cellular enzyme, it starts transcribing the positive sense RNA into a negative sense RNA. Once the negative sense RNA is ready to go, again the cellular enzymes does the transcription into various different RNA of the virus proteins like structural and accessory proteins. Then those RNAs will be translated by, again, the cellular enzymes into appropriate proteins, will be transported into their proper places such as Golgi and endoplasmic reticulum, where they get matured into a much more effective proteins, and they will start assembling the virus at the membrane of Golgi or membrane of endoplasmic reticulum. Once the virus starts assembling at the membrane, an exosome will be formed. Once the exosome formed, the exosome reaches to the cell membrane and the virus will be released into the bloodstream to into the outside of the cell to infect the other cells and increase their population more and more. Entire this process, exit process, is called exocytosis. Like I said in the beginning, they do not divide 1 to 2 or 2 to 4 or 16. This is entire process is like an assembly process. It's completely like assembling a car. All the body parts are manufactured various different places of the world, will be shipped into one place, and then assembly will start taking place in one place, and a functional car will be prepared and made at that place. So once virus start infecting the human cells and it releases the new viruses into the infect the other cells, the very basic defensive mechanism, defensive system activated in the body is immune system. in human immune system has two different types of immune system. One is innate immunity and second one is adaptive immunity. The very first immunity that responds without non-specifically, which is called a frontline defense, to react or fight with the virus or any other infections. And the innate immunity activated within hours. And also another important point here is this: every human being contains the innate immunity by birth, but adaptive immunity doesn't come with birth. This is like a backup defense. It is very specifically reacted. It requires an antigen molecules specifically to respond. But once adaptive immunity is activated, it lasts for longer times, months to years. It's slowly activated. It takes days to activate. However, there will be always a crosstalk between innate and adaptive immunity systems. And both the immune systems contain various different cell types that I'm going to explain you in a minute. Excuse me. So the innate immune system, which has many different cell types, but I'm going to talk about three major cell types here today. Very first thing, as soon as it is activated, is it releases various different signaling molecules, which are called cytokines and chemokines, proteins and chemicals. These molecules will reach to the infection site and attaches to the infections like viruses and infection cells to generate the signals. Once the signals are generated, macrophages will be activated. These macrophages, basically finding the signals, they go to the virus and find the viruses, they engulf the virus, they eat it to clear out from the body system. While macrophages is doing their job, nature killer cells start acting. They initiate the suicidal mechanisms in the virus infected cells so that they can be killed themselves. Once the cells are dead, the macrophages will come and eats them up, even the infected cells, the infected dead cells, to clear them out from the body. While macrophages and nature killers are doing their job, antigen presenting cells, they start collecting the antigen molecules from the viruses and virus infected cells and take those antigen molecules and runs towards the adaptive immune system to activate the adaptive immune system. Adaptive immune system contains two different cell types, T cells and B cells. Once they get the antigen molecules from the antigen presenting cells, B cells immediately releases the plasma cells and various different types of antibodies, including the neutralizing antibodies. These antibodies, neutralizing antibodies, they go and bind to the spike protein of the coronavirus so that they inhibit the interactions between ACE2 receptor and spike protein so that they cannot infect the new cells, so that once they basically arrest the virus, once the virus is arrested by the neutralizing antibodies, macrophages will come and eat them to clear them out. And T cells will be divided into two different cell types, cytotoxic T cells and T helper cells. The cytotoxic T cells are kind of a additional force to the nature killer cells. They do the same job. They activate the suicidal mechanisms, they eat up the cells, they kills the cells. Macrophages, they clears out these cells as well. But the T helper cells generates the memory cells. These are the very important cells in the adaptive immune system. So these memory cells, what they do is they remember the characteristic features, the antigen molecules on that particular virus, and they remember they also remember the fighting mechanism, how to clear out the virus. And whenever the new they remain active for months to years. And whenever the virus infection comes into the body, they will clear the virus infections out because they remember their antigen molecules and their their clearing mechanism as well. However, the coronavirus somehow escaping from this immune system attack. There are various different. Can you please mute someone? It's very disturbing. Whoever is speaking, please stop. Okay. All right. So the coronavirus escapes from the immune system somehow. There are various different approaches are available that I'm going to explain in a minute. But the very first thing the coronavirus, as soon as it infects the human body, is it diverts the immune system. It sends the wrong signals to the immune system. The immune system immediately releases a storm of cytokine and chemokine chemicals, which is called, process is called cytokine storm. When these cytokines are also two different types, inflammatory pro-inflammatory cytokines and anti-inflammatory cytokines. When the pro-inflammatory cytokines are released in a larger amounts, they will start damaging the tissue in the organs. As soon as the tissue is damaged, the organs will start malfunctioning. When the organ is started malfunctioning, it will go into the death, organ death, and multiple organ death causes the death of the patient. This is how it's acting on the infected patients, leading to the death. There are various different hypotheses are available at the moment. How the coronavirus escapes from the immune mechanism. There are some evidence is saying that accessory proteins probably helping the coronavirus to escape from the immune attack, so that they will not be attacked by the innate or adaptive immune system to clear out from the body. The second hypothesis is the conformational flexibility of spike proteins. This is the cryo-electron microscopy structure of the spike protein, which has a receptor binding domain here. This is an active form. In active form, it is located as a linearly, which effectively binds to the ACE2 receptor to infect the other cell. And the second inactive conformation, where it linearly oriented, which does not bind to the ACE2 receptor. So when immune system is trying to find a spike protein or coronavirus, it is into this inactive conformation to escape from the immune system attack. And the third hypothesis here is the sugar coating on the spike proteins. Glycosylation is another concept which provides the stability, solubility, and interaction flexibility to the various different proteins. So glycosylation is nothing but bunch of sugar molecules together decorate the proteins. So spike protein has about 22 to 27 sites that can be glycosylated. This is also another reason. These are very well decorated so that the immune system cannot identify the spike protein to escape the virus from the immune system. The recently, couple of weeks ago, this new study has been published that the NSP1, non-structural protein 1 from the coronavirus, inhibiting the protein synthesis of immune system proteins like anti-inflammatory cytokines or some others. So what it does is, and on a ribosome, in generally, there is a tunnel where mRNA will be entered and it will be translated into the protein and released into the cytoplasm or endoplasmic reticulum to send it to the appropriate places. But this NSP1 binds to the tunnel, blocks the tunnel to inhibit the translation of the mRNA, so that the immune system cannot actively create the chemicals or cytokine molecules to kill the virus or inhibit the virus. This is another mechanism that has been proposed to that the coronavirus escaping from this. So when we have this coronavirus escaping from the immune system and it unable to control by the own body, then this is when we needed the external force, extra molecules like vaccine or therapeutic developments. Now I'm going to talk about. So first I'm going to talk about the vaccine development process. Then I'm going to come into the antiviral therapeutic development process. So the most important thing about the vaccine is vaccine doesn't cure the disease. It can prevent the disease or it can prevent the infection before it comes into the human body, like a virus or bacteria or toxin or anything else. That's when, but the virus already infected, then we need the treatment options such as developing antiviral drugs or therapeutic molecules. So before I go into the vaccine developments, let's take a look at how does a vaccine works. Vaccine in generally strengthens the adaptive immune systems. When a vaccine is given into the human body, administered into the human body, vaccine is nothing but a virus or virus-like a molecule or virus-like a particle or a part of the virus or a protein or a nucleic acid of a virus. When it is injected, the immune system will be responded. The adaptive immune system thinks that the virus came inside the body. Then the antigen-presenting cells collects the antigen molecules and gives to the adaptive immune system, which activates the T cells and B cells. B cells releases the neutralizing antibodies and various different types of antibodies. And T cells generates the memory cells, which clearly remembers the antigen molecules and killing mechanism of that particular virus and remains as a rapid force or a defensive force when the real virus comes into the body to clear them out from the body. So the virus or vaccine or potential therapeutic development has two different stages, preclinical stage and a clinical stage. In preclinical stage, the vaccine or a drug molecule is developed in the lab, created in the lab and tested in the mammalian cells first. Once it is passes that stage, it will be tested in the animals. Once the safety levels are cleared in the animals, it will be, if possible, will be tested couple of healthy individuals at preclinical stage if they find any healthy volunteers. Otherwise, it will be moved into the clinical stage. In clinical stage, there are 4 different phases: phase 1, 2, 3, and 4. In phase 1, there are less than 100 healthy individuals will be tested for safety and dosage of a vaccine or a drug molecule candidate. Approximately 70% of the candidates will pass from phase 1 to phase 2. In phase 2, more than 100 people, up to a couple of hundred, 200 to 300 people will be tested for the efficiency of the vaccine candidate or the drug candidate, and also they test for any severe side effects. And approximately 33% of the candidates will pass through phase 2. In the phase 1 and 2, only healthy individuals will be tested for any potential drug between the age of 18 to 55. And phase 3 is the most important phase, where thousands and thousands of people will be tested for the safety, efficiency, and side effects, adverse reactions, not only in the healthy individuals between 18 to 55 years age, but also more than 55 years age, older people, as well as people with conditions, pre-existing conditions such as diabetic patients, heart patients, or dialysis patients, etc., will be tested for the safety, efficacy. Then, at this stage, the regulatory committees will decide whether it can be released into the market as investigational new drug or new vaccine or emergency vaccine. Once it is released into the market, still this is called a phase 4, and it will be continuously monitored in the real world for the to check the long-term side effects. Approximately 25% candidates will be passed from phase 3 to phase 4, and less than 10% will be passed from phase 4 and clears out complete clearance to as a vaccine or a drug candidate. So in creating a vaccine, there are various different methods or approached used to develop vaccines for the coronavirus, present SARS coronavirus 2. To develop vaccines, the most important molecules from the virus are antigen molecules are very important. In SARS coronavirus 2 case, structural proteins such as matrix, nucleocapsid, envelope, and spike protein. Spike is the most important protein, plays a very important role in developing the vaccines. As of today, according to the World Health Organization, approximately 176 potential vaccine candidates are under development, and 34 of them are presently in human clinical trials. So among various different methods, I'm going to go through one by one different approaches. The first approach is using the viruses themselves as a vaccine. There are 2 methods in here using viruses to develop potential vaccines. First method is live attenuated virus or weakened virus. They take the original, let's say coronavirus, take the coronavirus, weaken it using various different methods. For example, passaging them in human cells or animals until it gets weak, because viruses mutates constantly. It could be a bad mutations or good mutations in the sense of virus. When bad mutations happens to the virus, it weakens the virus capacity. When good mutations happens to the virus, it becomes more and more stronger. So these are the examples: smallpox vaccine, measles, mumps, rubella, MMR vaccine, chickenpox, influenza vaccines are successfully developed using this live attenuated method, are currently being used. At the moment, Indian Immunologicals, Serum Institute of India, Codagenics, these companies are presently using this method to develop vaccine against coronavirus. These are all at the moment in preclinical stage. Still, preclinical studies are ongoing. This is a bit time-consuming process. The second method is using the inactivated virus, killing the virus using certain chemicals like formaldehyde or heat. Heat-induced inactivation also is one of the popular, or egg albumin, using egg albumin to inactivate the viruses. So when the virus is inactivated, it cannot infect them. It can just act as a pseudo. Can also induce the immune response. The examples include polio virus vaccine, hepatitis A, rabies are successfully developed using this method and currently being used in the world. Sinovac, Sinovac, Dynavax, and Bharat Biotech are the companies, one of the front runner companies using this method to develop the vaccines. At the moment, they're all conducting human clinical trials when phase 1 and 2 clinical trials are ongoing. A second method is using the viral vectors to develop the vaccines. This approach is also has two different methods, which is using the replicating viral vectors. In this method, basically they use live attenuated virus. They engineer the virus, attenuated or weakened virus, or such as measles virus, and now recently people are using vesicular stomatitis virus also as one of the replicating viral vector. Engineer them so that, for example, for coronavirus case, they engineer the spike protein gene into this attenuated virus so that it can express the spike protein to induce the immune response. Second method is using non-replicating viral vectors. So these are all live viruses, but they don't replicate like a viruses. So amount of the virus is given inside the body will induce the immune response. They can be engineered, such as engineer the spike protein into this virus vector that can express the virus spike protein in the once enters into the human body, so to induce the immune response. Most commonly used vectors are adenoviruses, which are most commonly cause mild flu or cold in humans. This Consumer Biologics is the company that is far ahead in any of the companies developing the potential vaccine for coronavirus 2. They have used adenovirus, non-replicating adenovirus vector, a serotype 5, 85 vector. They engineered it to spike protein into it to express the spike protein gene in the when it is administered. So they have successfully completed. They went very rapid speed. When the virus infection was discovered in December 2019, by February they completed the preclinical studies, which usually takes approximately around 5 years. And entire this process, the preclinical to clinical process, takes approximately 10 to 12 years to develop a vaccine or a potential drug candidate. But because of the emergency now, lots of regulatory authorities are giving lots of parallel conduction studies. So many reliefs to the vaccine development companies. So that's why they were able to do much more faster vaccine development. So right now, phase 1-2 is ongoing for this particular vaccine candidate, and they also published interim data, which showed some good signs. In phase 1, they conducted conducted on 108 people. Phase 2, they're doing on 500 people. Currently, they're recruiting for thousands of people for phase 3. And entire this study was done only in China. This is a Chinese-based company. They have this vaccine in their report. They said it has a very good safety profile without any side adverse events occurred. These systemic reactions is common, like mild fever, fatigue, headache. When a vaccine is administered, a new vaccine is administered, if these small reactions are present for a couple of days, is fine. If they are beyond the couple of days, which is a serious adverse event. So that is not observed in this vaccine candidate, and it induced a rapid immune response within 2 weeks of the administration. They monitored the antibody production, T cell production, up to 4 weeks, 28 days. The second company, AstraZeneca, together with Oxford University, are developing another vaccine. They are using a chimpanzee adenovirus. This is also non-replicating virus vector. They have engineered spike protein into chimpanzee adenovirus vector. They have also successfully completed preclinical studies. Presently, they are conducting phase 1, 2, 3 simultaneously on thousands and thousands of patients. They also released interim data, which showed very good signs. 91% participants showed very good immune responses. They also monitored up to 56 days, approximately 2 months, that the antibodies produced are sustained and the T cells produced are durable. Their longevity is maintained. And good thing about this is they are conducting the clinical trials not only in the UK, but also various different countries, Brazil and South Africa as well. So their goal is to produce approximately 2 billion doses globally by end of next year. Another company, Janssen Pharmaceuticals, is a sister company of Johnson & Johnson. They're also using a non-replicating adenovirus vector, engineering the spike protein. This is a 26th serotype. Successfully completed a preclinical study. Currently, phase 1 and 2 is ongoing. They're also conducting in US and Belgium, multiple different countries. They're planning to extend their studies in Japan, Netherlands, Spain, and Germany as well. They also published interim data, showed a very decent, very good results. And their goal is to produce to about 1 billion doses globally by end of next year. And second approach is using the nucleic acids as a potential vaccine candidates to develop vaccines against the coronavirus. So this is the cryo-electron microscopy structure of the spike protein I have mentioned in my previous slides as well. And in this method, they can also use the only a receptor binding domain or only a particular subunit, S1 or S2 subunit, encoding either DNA or RNA to develop the potential vaccine candidate. In DNA in general, the DNA plasmid will be directly injected into the muscles to generate the immune response. But RNA need to be encapsulated into either a lipid nanoparticles or a formulation, certain type of formulation to inject into the human body. Moderna Therapeutics, this is a USA-based company. They are using, they're developing a mRNA-1273 vaccine. They're using RNA that encodes the entire spike protein in a pre-fusion form. They're encapsulating this mRNA into lipid nanoparticles and administered into the muscles intramuscularly. They completed the preclinical studies. Presently, 3 phases are simultaneously ongoing in multiple countries with multiple thousands of people. They also released interim data with very good results, monitored up to 2 months, with that produced neutralizing antibodies and T-cell responses as well. Their goal is to produce 500 millions to approximately 1 billion doses per year from next year onwards. The second company, BioNTech, a German-based company, together with Pfizer and Fosun Pharma from China, Pfizer from United States, developing another mRNA-based vaccine to fight with coronavirus 2 infections. They actually started with 4 different candidates, of one of them using unmodified RNA of the spike protein as it is, a modified RNA of the spike protein, 2 candidates, and another self-amplifying mRNA. Out of the 4, these 2 went into the clinical phase. Among those 2, BNT161B2 went is in further ahead in phase 3 clinical trials are being ongoing right now. They also published some interim data which showed a very good result with a good safety profile and good immunogenicity developed, monitored for 43 days that they sustained the antibodies produced and T cells generated. Presently, they're conducting the phase 2-3 simultaneously in USA, Argentina, Brazil, and Germany, various different countries. Their goal is to produce 100 millions to 1 billion doses per year from beginning of next year. Inovio Pharmaceuticals is another US-based company developing a vaccine which is called INO 4800. They are using DNA-based vaccine. They are using a plasmid that encodes the spike protein. So they also have a device developed. They can intradermally, using electroporation method, injected or administered in the skin, which even faster spreads into the, activates the immune response. So they successfully completed a preclinical studies. Presently, phase 1 and 2 are ongoing. They're all conducting in multiple countries like US and Korea. And one important thing is this is the only company presently have a vaccine candidate. It's phase 2 clinical trials for MERS coronavirus, another coronavirus that I mentioned in my previous slides. The other approach is using the proteins to develop the potential vaccine candidates. In protein-based methods, either spike protein, part of the spike protein will be directly injected using formulations. So Novavax is the company that using the spike protein subunits and entire spike protein into lipid nanoparticles to develop the potential candidate to fight with this coronavirus. They successfully completed preclinical. Right now, phase 1-2 is ongoing. They also released interim data up to 4 weeks. Sustainable results were obtained in 100% of the participants. Phase 1 and 2 is also going on multiple countries, including US and Australia. So now I'm going to talk about the therapeutic developments, what's going on. So in therapeutic developments, there are two methods are approached. Two approaches are developed. One is repurposing the existing therapeutics. Second one is developing the new targeting drugs from the scratch. In repurposing the therapeutics, the existing broad spectrum of antiviral agents could be, or maybe any other drug molecules will be screened against this coronavirus to find the potential therapeutic molecules. Second one is the developing the drugs from the scratch, which is very time consuming. So that's why the first method is much more rapid. So I'm going to give you some examples in my upcoming slides. As of today, approximately 379 potential therapeutic agents are under development. 293 are already in human clinical trials. So these are the candidates that are identified in drug repurposing, which antiviral already approved by FDA and CDGI in India for the various virus infections. I'm not going to go into the details because we are almost running out of the time. My apologies for taking extra time. I'll be wrapping up in a couple of minutes. So all of these are targeting particular proteins of the coronavirus. And the remdesivir has been already approved as an emergency use, already being used in the people all over the world. And also favipiravir also approved as an emergency use to treat with patients with mild infections. This is to treat with patients with severe infections. And these three also are presently are being tested in various different patients. Favipiravir, as I mentioned, this is the structure of the prodrug. Once it is enters into the body, it will be phosphorylated using the body enzymes. This is the active form of the drug. Right now, it's in the phase 3 clinical trials. As I mentioned, it's been approved already, and also in India, it's been marketed by multiple companies. This enzyme targets the replicase enzyme, RNA-dependent RNA polymerase, to inhibit the virus replication of coronavirus infections. Remdesivir also targets the replicase. Multiple companies are marketing this in India and US, all over the world. This is the structure of the prodrug, which will be converted into active drug. Is a mimic of adenosine, which is a nucleotide that makes the nucleic acids such as DNA and RNA, which is currently in phase 3 clinical trials. This molecule actually potentially being developed to control the infections of Ebola viruses. But when it is repurposed, it showed a very good promising results towards the coronavirus. That's why it's been in the clinical phases, and now anytime it can be approved, it will be approved as a full drug for the to treat the coronavirus patients. And this is a crystal structure where it showing the inhibition mechanism of the replicase. This is another molecule collaboratively being developed by Emory University in the US, Ridgeback Bio, a biotech company, and Merck Pharmaceuticals. This is called EIDD2801. This can be taken as an oral pill. This molecule also is being developed to protection from Ebola virus infections. But when it is repurposed against coronavirus, it showed promising results. Presently, phase 1-2 studies are ongoing. Hopefully, this will be soon approved to protect people from coronavirus infections. And this is another method, hyperimmune therapies, using the neutralizing antibodies, another method to prevent the coronavirus infections, which my lab is working on. Unfortunately, I cannot share the data because of the confidentiality and conflict of interest issues. But I can give you a glimpse of what we are working on, how we are doing this kind of research. So as I mentioned, this is the cryo-electron microscopy structures. To inhibit the virus infection, we need to find a very efficient antibodies that can inhibit the receptor binding domain, binding to the ACE2 receptor. So this is the crystal structure. This is the antibody that is isolated from the convalescent patients. It is binding to the RBD, but it is not binding where it's supposed to bind to inhibit the interaction between ACE2 and RBD. So what we do is we take these type of antibodies isolated from the convalescent patients, use some machine learning computational algorithms, make them better, synthesize them in the lab, and test them in the pseudovirus, then test them in the real virus. And this is how we are trying to develop potential hyperimmune therapies in my lab. So far, I have discussed about the evolution of coronavirus towards the COVID-19 pandemic, and how the body surveillance system, immune system protects us from virus or any other toxin infections, how even virus escapes from these immune protection systems. And however, and also I have discussed about the potential developments in the vaccine and therapeutic molecules of the coronavirus to COVID-19 infections. However, as I mentioned, among the 7 serotypes of the coronaviruses, 3 of them are very dangerous, much more lethal, pathogenic. SARS-1, SARS-2, and MERS-2. All these 3 coronaviruses till date, this SARS-1 was existing from 18 years, MERS existing from 8 years. Still, we don't have any vaccine to protect from these 2 infections. They may come back, and also SARS-2 coronavirus. So how can we protect from the infections? Here is the slide. This mostly the chemistry students can understand better because these cartoons are much more useful and informative to the chemistry students. So social distancing is one of the important. Lots of people are saying social distancing is important. Stay home unless until you need to go out. And to strengthen your immune system is also very important. Most important thing is you have to live like you're living with the virus in your house. That's what is, you have to take it. And strengthen your immune system by, eating a very proteinaceous or, healthy food, healthy diet, nutritional food. Get some sufficient sleep. Be active. Get some active work style. Exercise like that, so that your immune system will be strengthened. Particularly adaptive immune system to protect you from virus infections. Even though it is coming, you are infected with the virus, the immune system fights much more effectively to clear it out from the body. So in my journey started from OU campus at Department of Chemistry. On OU campus, I started learning chemistry from there. Then I briefly worked at CSR IISCT a couple of years where my research experience started. Then I moved to South Carolina Department of Chemistry and Biochemistry for my PhD, where I successfully completed in organic chemistry. Then I moved to Wayne State University for my postdoctoral studies, where I learned how to work with bacteria while I'm exploring my synthetic skills. Then I moved to MIT for my first job, where I started working with human cells to study the various different cancers while I'm exploring my skills that I have learned at South Carolina and Wayne State University. At presently, I'm working at Expansion Technologies. Right now, we are developing various early stage diagnostic methods to detect the cancers in early stage. However, since the pandemic started, we take up the antibody therapeutic project, and actively we are working on developing novel antibodies to fight with the coronavirus infections. So I would like to take this time to thank all the people who helped in this my journey, my students, my mentors, my teachers, my gurus, my students, my colleagues, my friends. Without their help, including my family members, without their help, I don't think I would have been what I am today and where I am today. Having said that, thank you so very much for your patience. I appreciate your time. I apologize for taking 7 minutes longer, but I'm happy to take any questions if you have.
Thank you, sir. Thank you for this informative session. So here are a few questions asked by our students.
Sure.
Can humans become infected with COVID-19 from a food source?
Yeah, of course, possibly. It depends on what type of food, right? If the food is contaminated with the virus, obviously it can infect the the person.
Okay. Okay, sir. So moving on to the next question.
Sure.
What are the treatments available for COVID-19?
At the moment, the treatment options, right? Remdesivir is one of the treatment. Favipiravir, second one. So remdesivir can treat severely infected patients, which can cure the infection within like 10 to 11 days. Which again, this is an emergency medicine. If it is really required, can be administered only. It's not like a common medicine. Any patient can be taken. It has to be taken by a particular prescription or doctor recommendation only. The second one is favipiravir. This can be administered to the people or patients with mild symptoms, and this is also approved only in emergency case from CDGI, Drug Controller General, DCGI, Drug Controller General of India, or FDA in the USA.
Sir, how about this hydrochloroquine?
Hydroxychloroquine is it's a malaria medicine. In the beginning, back in the days, like about 2-3 months ago, it was reducing the symptoms like fever, cold, cough. But later on, lots of companies started clinical trials using the hydroxychloroquine. But its side effects are much more severe than the curing effects. So that's why lots of companies they just stopped. And now I don't think it's being used to cure or treat any patients.
Yes, sir. Now Dr. Ashok wants to ask a question. Please go on, Professor Ashok.
Sure.
Professor Ashok, we are unable to hear you.
Dr. Mahender Dewal.
Yes.
Yes, sir.
Your talk is very excellent, highly informative, useful, and definitely it is useful to many young people who are attending. Thank you very much. But I have one question.
My pleasure.
I have one question.
Sure.
Recently, there were 2 contradictory statements by USA and WHO.
Yeah.
USA says that vaccine will be available by first week of November 2020. But the WHO says that it is not possible to get new vaccine for COVID-19 before middle of 2021. As a layman, which we have to understand and which is practically possible.
Okay, yeah, that's a very good question. So what WHO said is correct, 100% true. And the November 1st availability in the US is not spoken by any scientist or any doctor. So I do not believe that at all. And also, I mean, even the whoever is attending this talk, I would like to mention that the vaccine. I already explained the process, the development process. It takes months to years. So do not believe that the vaccine is coming November or August or December. It may be approved. They may be, in my opinion, among all the vaccine potential candidates that I presented, approximately 3 to 5 vaccine candidates could be available by next June. And maybe approved as emergency or anything else. But in December, maybe 1 or 2 of them can be approved as an emergency use, but it may not be available to the entire population of the world because the production is going to take even months and months. It's impossible. A common man to get a vaccine might probably end of next year. That's the only hope I can see, or June, July. Even see, one more important thing we need to think about is the longevity. So we only have data up to 2 months. Yes, antibodies are developed when the vaccine is given, and the T cells memory cells are generated. But how long they are sustained, how long they are stayed active, we don't know. So at least we need 1 year of time to see that data. So that's all the companies started their clinical trials in May. So that means next May to June is the time that I can confidently say, yes, this is a good vaccine, this is not a good vaccine. And we can have the vaccine available or not. I hope I answered that question, sir.
Oh, thank you very much. Dr. Sarasita would like to ask one question.
Please go ahead.
Good morning, Mahender Dewal ji.
Yes, ma'am. Good morning.
Your lecture is excellent. Really, it is very inspiring and present pandemic situation suitable. It is very fine lecture. And I have a question. When this COVID-19 can end?
Excellent question. So the COVID-19, it's, I mean, we cannot say that it's going to be in tomorrow, or nobody can give you a certain date. So the COVID-19 pandemic probably it started going down, slowly declining. It all depends on us, basically, how we protect ourselves. So if you clear out the virus, let's say the most important thing in here is more testings need to be done, more contact tracing need to be done. When you trace the contact and hold him in one place, and you can stop the spread of the virus. As you stop the spread of the virus, then that's it. The virus is going away. So no more new infections are coming. That means the virus infections are going down. As soon as you see the decline, that means the COVID-19 is going down. But when that's going to happen, it all depends on by country, by people, by environment. Various different factors are going to influence this decision. So my goal, my opinion, my prediction would be sometime next year, maybe end. But the situation is going to be better, definitely. By end of this December, we're going to have a much better condition, better situations. It may not be the international travels or the traveling may be restricted, but the living style, lifestyle will be definitely improved. It's not going to be as bad as a couple of months ago or at the moment.
Thank you.
Sir?
Yes.
Good morning, sir.
Good morning.
If a person if a patient is cured from this COVID disease, again, is there any chance to reoccur?
Good question. Very good question. It all depends on the patient. See, 90% is not possible. In general, reinfection is kind of rare because the body already produces the memory cells, right? The immune system responds successfully clear the virus. Memory cells are already there, antibodies are already there. Unless until the patient or the infected person hasn't developed a very strong immune system. And he already has some kind of pre-existing condition like diabetes or anything else. The immune system hasn't developed a very strong immune response. There's a possibility that, but in very rare cases. I hope I answered your question.
Thank you, sir.
Sir, excuse me, sir. I am Rajini.
Yes, go ahead.
It was a very enlightening lecture, sir. I'm very happy to have you, and sorry also on behalf of us, we have made you wait for such a long time.
No worries.
But we had a beautiful lecture we had, and how the process is going on, everything is very clearly explained, sir. And I'm happy that I could hear something from you that by next year we could have something, or some medicine or a drug or a vaccine. That's good. But since the strains are going on changing, the structure is being changing, they are telling, does the vaccine can be used for all types of strains, or we should go on continue our research?
Yes, so it all depends. Yeah, very good question. Excellent question. It all depends on how rapidly. See, for example, I have explained 7 different serotypes, right? SARS coronavirus 1 vaccine may not be working for coronavirus 2 vaccine, but if the coronavirus 1 mutated to a different form, but the mutations are not going to be heavily impacted. Generally speaking, there is a possibility, is very low percentage possibility. And also, most of the vaccine candidates at the moment are also testing the mutation that is happened from March, which is D614G that I mentioned in my slides. And all the vaccine, most of the vaccine, not all, majority of the vaccine candidates are also effective in that mutated virus as well. So, and another good point here is some companies trying to develop a vaccine, pan-coronavirus vaccine. They're calling pan-coronavirus vaccine or pan-coronavirus medicine, which can protect from any coronavirus, any serotype of coronavirus. Let's hope they may get success. The success rate is kind of low, but you never know. This is a drug development process, right?
Thank you, sir. It gives a positive.
Yes, I hope you answered the question.
Yeah, sir.
Thank you,
sir. I'm Dr. Swarupa. I have a question.
Yes,
sir.
Dr. Swarupa.
Some of the reports are saying that the virus is present in fecal matter for about a month, sir. Sir, is there any chance for transmission from this fecal matter, sir?
Fecal matter, right? It's a very good question. We do not have any data saying that it transmits from mother to child. In fact, not even from mother breastfeeding as well. So the chances are very, very, very low. We do not have any, because this is a respiratory virus. So respiratory virus mostly majority transmits through respiratory fluids, respiratory droplets. So not even it transmits through blood. So the chances are low. I hope I answered your question.
Okay, sir. Thank you, sir. Thank you.
Thank you very much, sir. I think Dr. Raji Reddy garu is waiting.
Sure.
Now I request Swarupa to present a memento to our Dr. Mahender Dewal garu. Please, Swarupa.
Yes, ma'am. Yes, ma'am. Good morning, everyone. It's been an honor for us to have Dr. Mahender Dewal garu with us today. We thank you, sir, for starming our brains to think differently with regard to the present COVID-19 pandemic. Thank you, sir, for giving us a detailed view into COVID, the viral structure, and the progressive infection and pathogenesis. All of us must have understood that there are various strains of coronavirus, and the current pandemic is caused by a novel COVID strain. I, being a microbiology faculty, express my thanks to you for sharing such a comprehensive session, especially on the course of infection, including the endocytosis, replication, assembly of the viruses. Also, the immunity towards the virus. Your talk has been very student friendly, and I hope our students have imbibed this detailed information about COVID. Sir, you have given a clear picture on the recent developments of vaccines and repurposing of therapeutics. I thank you for this detailed information, sir. And one more thing, sir. Recently, I was fascinated by your interview on Q News, wherein I came to know that you are alumni of Giraj Government College, Nizamabad. Coincidentally, I too have graduated from the same college. And I would like to reproduce some of the words spoken by you in that program, sir. You said, you said and proved, the language is not a barrier, whether it is English medium or Telugu medium, to aspire high. Just believe in yourself and be confident. I hope our students also get inspired by your words and believe in themselves. Thank you, sir, for giving me this opportunity. Thank you.
It's my pleasure. Thank you so much for your kind words. And also, I mean, if I haven't answered any, there may be many questions. Whoever has any additional questions can reach out to me on Facebook or Twitter. I'm happy to answer your, clear your doubts question, because lots of people have lots of doubts, and, lots of problems as well. So feel free. Thank you so much for hosting me.
Thank you, sir. Thank you,
Thank you,
Swarupa,
Dr.
for
Mahender.
yeah.
Thank you, Dr. Mahender.
My pleasure, sir.
Thank you. All the very best.
Yeah. Thank you, Swarupa, for presenting memento to Dr. Mahender Dewal garu, and then presenting an appreciation certificate also. And once again, from the bottom of our hearts, we thank you, sir, Mahender Dewal garu. We hope to see you once again somewhere.
Sure. My pleasure, certainly. Please feel free to reach out to me.
Thank you very much, sir. Thank you. Now, may I call upon Sumati Natarajan to introduce our second resource person, that is Dr. Raji Reddy garu from IICT. Please, Dr. Sumati Natarajan.
Yes, ma'am. Thank you, ma'am. Am I audible, ma'am? Hello?
Yes, yes.
Thank you, ma'am. Thank you, sir. Dewal, sir, it was a very, very wonderful session. Very, very detailed explanation. And now we are intrigued more into the science of pharmaceutical and medicinal chemistry. And today we have our next speaker, who is a principal scientist from the Department of Organic Synthesis and Process Chemistry at the Indian Institute of Chemical Technology, Dr. Raji Reddy garu. Sir and his research team have been actively involved in synthesizing novel chemical entities on the principles of green chemistry that can be used as anti-cancer therapeutic agents. Dr. Raji Reddy completed his graduation from Kakatiya University and post-graduation in organic chemistry from Osmania University. His interest in organic chemistry drove him to pursue research and complete his doctoral dissertation on anti-cancer compounds, epothelone, at IICT. And in 2002, he joined as a doctoral fellow at University of South Florida. In the same year, he was awarded with postdoctoral fellowship by Center for Disease Control to follow his interest further in the organic synthesis in the University of Mississippi. In 2005, he returned to India to be subsumed as a principal scientist at Sai Life Sciences Private Limited at Hyderabad. From 2006, he has been a part of IICT and is presently the principal scientist in the division of Natural Products Chemistry. I would like to share a few of his research contributions to medicinal chemistry. Sir has synthesized the pentasaccharide, which is a precursor for an antithrombic agent, in collaboration with Bharat Biotech. He was associated with Volva Biotech Switzerland to synthesize and screen various antifungal agents against Candida, one of which is also under phase clinical trials. Sir is a recipient of numerous national and international awards, including the Bronze Medal Award by the Chemical Research Society of India. It gives me immense pleasure to invite Dr. Raji Reddy to deliver this talk. This session is all yours, sir. Thank you.
Dr. Raji Reddy garu, really sorry, sir, for making you to wait for such a long time. Please take over, sir. Dr. Raji Reddy garu, please take it.
Thank you, madam. I thank Dr. Sumati for her kind introduction words. Before starting my lecture, I would like to thank the Telangana Social Welfare Residential Educational Institution Society for inviting me to deliver this talk, and in particularly Professor Rajni Madam and Professor Ashok, who is my teacher, taught me initial organic chemistry basics. From there, I have grown up till in today's present position. So I also like to thank the secretary of this society, Praveen, and joint secretary also happen to be Praveen, both of them, for their interest to bring the students into the international competitive level through the society. It's a really wonderful job. I used to hear a lot about Praveen sir IPS, his contributions to the society through media and several other newspapers and other things. So with this, I would like to start the lecture. I would like to share the presentation. Give me a second, please. Yeah, hope you are able to see this.
Yes, sir, clear.
Okay. So what I will try to do, Dr. Mahender, he already mentioned, he was my classmate during masters. We stayed in Usmania University B hostel on the same corridor we used to stay, and we had a good time. And he gave very vast introduction about coronavirus, how it got developed, what are the different coronavirus previously, and many things. What I'm going to do here, keeping the graduation students in mind, I thought how chemistry plays a key role in drug discovery in general, but in particular, how it played a key role under this pandemic situation towards COVID-19, either it may be prevention or maybe treatment or maybe detection, any thing you take, there is a chemistry involved, so how it plays a key role. So initially what I thought was, since there are a lot of graduation students are there, how chemistry plays a role in drug discovery, how it will be done. General process I thought of giving, focusing organic synthesis as a key point, because what kind of role organic synthesis plays in this drug discovery or when the drug wants to come into the market, all these points are involved mainly from organic synthesis. So I thought of, and what is process chemistry? So I want to cover a overall view. You may not, you don't need to really think of chemistry, what I am showing here. I will not discuss a technical points, but I would like to give a brief overview of this process. So before start my lecture, I would like to dedicate this lecture to all my beloved teachers who taught me from the day one, from our initial education onwards till today, all government schools and colleges, at Jilla Parishad school at my village, and then later Government Junior College, and followed by SRR College, Degree College, Karimnagar, and then Usmania University, PG College, and later IICT for my PhD. I thank all my teachers who made me today at this stage. So with this, I would like to begin the lecture. What I am going to cover in my lecture is mainly, I will not go much details about coronavirus and COVID-19. Dr. Mahender already gave a vast points and covered this, but I would like to cover the drug discovery, how organic synthesis plays a role, and what is our experience at IICT, what we are doing, and what are our contributions towards this drug repurposing, and then little bit process chemistry. Finally, one slide overall, what CSR contributing towards COVID-19. So these are the contents I would like to cover. So to just recap once again, I think we all know that in early 2020, this new type of virus was announced by WHO. Then it was mainly spreads from person to person, and some cases proved that it's touching a surface or object. And then the disease caused by this virus given a name as a COVID-19, coronavirus induced disease. And then these are the symptoms. So what we are doing for preventing this, all of you know that mainly frequent washing our hands, and if the water or soap not available, then we are using sanitizer and taking distance and other wearing mask and many other precautions. So now coming to the chemistry, how chemistry plays role in our life. Basically, chemistry is a central science, we all believe, and it related to each and every branch of science. You see here, this is mainly for students, undergraduate students. You imagine anything around you. Since morning, you start with brush and toothpaste. From there till you go to your bed, you use all out or whatever, and everywhere chemistry is involved, chemical material is involved, and you cannot avoid chemical. For example, if you simply take, best example is water. Without drinking water, you cannot live, right? So water is also a chemical. It has a formula H2O. You have hydrogen and oxygen element. So from there, if you start thinking of chemistry in each and every our activity or our whatever objects we are using in our daily life, without chemistry, you cannot imagine our life or any item or any object what we are using. So overall, you can just say that this is a general one. I made it so it's a chemistry means it's maybe health, community. You think of health, you need medicines, again chemistry involved. You think of environment, energy, you have batteries, again made up of with chemistry and chemicals. Medicines, industry and science, teaching, technology, you, me, everything is a chemistry. We can see the chemistry inside all these things. So that is why chemistry is everywhere. You can think of good or bad. You want explosives, again chemistry involved. You want to cure disease, again chemistry is involved. You want to clean your house and everything, again you need chemical. So you cannot live without chemical. Just that is what I want to convey here. That is where the chemistry plays a key role. This is a little bit role of chemistry towards this coronavirus. So you need the testing kits, initially reagents, standards. You need to extract RNA. This is again major chemistry role. You have to have hand wash solutions, sanitizers, all these again chemicals. Vaccines, as Dr. Mahender clearly mentioned, there are more than 100 vaccine projects in various phases of development all over the world. Some of them are advanced stage under phase 2, phase 1 clinical trials, and some of them are at the beginning stages. The other one is treatment of COVID-19. So you need pharmaceutical. This is what I am going to cover as a major in this lecture. So some of you may are thinking that why it is taking so long. Now it is close to 8, 9, 10 months and no drug is available. Sometimes, we hear that what scientists are doing, and so much technology developed, we say always, but there is no proper treatment for these things. So if you see how a drug discovered in olden days, 100 years ago, when we don't have proper medicines for most of the diseases, these are the treatments our ancient used to use, traditional medicines, traditional remedies. So these are the basis for our drug discovery. 100, 150 years ago, we used to use our grandparents have a lot of knowledge on this. If something happened, okay, when we got hurt, something, you give little bit haldi powder, and then like that we have a lot of traditional remedies, traditional oils from leaves we use. So these are the basis for drug discovery. So from nature, right? If you see, everything is from a natural product. Everything is available in nature. So you can divide into 4 sources from these natural products. One is microbial world. This is basically, cephalosporium, acremonium. Like this, some species also produces some chemicals. Once you isolate that chemical, you can check the activity of that particular chemical produced by the nature. Other source is plant. All of us know. Best example is Taxol, used for the treatment of breast cancer. This is from Taxus brevifolia tree. It gives paclitaxel, and small modification gave the drug. Again, nature is inspired for this development of this drug. We have marine sources. Lot of, today if you take lot of anti-cancer drugs are from marine natural products. These sponges and other things, they are under the water into the sea. So the natural product scientists will take these natural products from underwater and they extract and see what kind of chemicals are available, whether are they active for any disease. Then other source is animal sources. They also produces some chemicals, but there are very few who are isolated from animal sources. And then best example is one of the epibatid in African clawed frog. From skin, they have isolated this compound. So from these 4 types of, mainly from these 4 types of natural products, our ancient civilizations, mainly Chinese, Indians and North Africans, we have some written evidences about these natural sources, how they are curing various diseases. So then you can see this is the oldest drug, natural product as a drug directly used, morphine, for the treatment of malaria. This is the best history. You can see that it was written in the Greeks, 3rd century before Christ, and then Arab physicians were used, and this was used for various other diseases, asthma, bad eyesight, diarrhea, and several things. So these were actually initially isolated in 1806 as a crystalline pure substance from tree morphine called opium, that is named as a morphine. So that's why this compound given name as a morphine. Then slowly you can see, all of you know that, I think many of you know the aspirin, how it was discovered, hydroxysalicylic acid. It is again from a tree. By derivatizing one of this hydroxy group as a acetyl, it became a drug. Aspirin, all of you know, many people used to take, but recently due to some side effects, it has not been sold very frequently in all these shops. And then you can see other medicines, morphine, digitoxin, quinine, pilocarpine. Like this, you name old, and many of the old drugs are originated from the natural products. Little more complex structures. Don't worry about these chemical structures and these things. But You just think how nature gave us medicines. You take penicillin, narcoticin, and other natural products in this slide are vancomycin, erythromycin. You name these drugs, all are from natural products. The basis source is natural product. Little further, we have doxorubicin, best anti-cancer agent used for treating leukemia, and you have Taxol. Just now I told. Again, this is the tree. We isolated Taxus bravifolia tree, and then we modified here as a side chain, and then it became drug as a paclitaxel. So this is the history. You know, artemisinin, another drug recently approved for anti-malarial, and then this big structure you can see, helicandrin B, is the latest drug available for anti-cancer, particularly breast cancer. And this is helicandrin B is the key basis for the development of ergoline. At Harvard University, Professor Kishi wants to make this compound in the laboratory. Then while making, he made only right portion of this compound. You can see the right hand structure is exactly matching with this one. And when he was halfway of this molecule, he wanted to test whether this particular half fragment is useful for any other diseases. Then they found it is used for treatment for the breast cancer, and after several years it was launched in the market presently by Eisai Pharmaceuticals, and this is the best latest available drug, but still it is very expensive. IICT also worked on this synthesis of this molecule. So now you learn how drug discovery was started olden days, and then how in the present days how complex molecules we are making. Then what is this development process involves? So of course nature is giving compounds, but can we just take directly? No. It has to undergo several various number of processes. The initial, here I have given a brief idea how it works, a drug discovery process. So initially you need to identify target and validate, like which target the molecule which we are chosen is going to work. Then we have to screen variety of compounds, variety of their analogs, because sometimes you get a lot of problems, then you little bit modify. So you have to make almost more than 5,000, 6,000 compounds you have to screen. Then you optimize the lead. You found that at least 2 or 3 compounds, okay, they are the best among them. Then you study structure activity relations, and there you modify a little bit for improving the solubility or bioavailability, maybe safety issue, maybe they are affecting other organisms. So you need to look at all these late stage preclinical trials. Then once you found at this stage, normally people will go for 3 to 5 compounds they identify. Then it goes to clinical development in 3 phases. Phase 1, phase 2, phase 3. You need to study the stability. All of you know that each tablet will have an expiry date. Some tablets are only 6 months, some tablet 1 year, some tablets you can see 3 years. So that much stability studies have done. That tablet is in safe form for that many years. So all these things before decide, it needs to be studied in that manner. So finally, one compound will come into the market. It gets market approval mainly by FDA, Food and Drug Administrative, which is there in USA. So all this process, it's not easy as we discussed in 1 or 2 minutes here. It will take at least 10 to 15 years after identifying the disease. Then you should know the mechanism of the disease. Then you try to find which structure. All these things will take 10 to 15 years. One is this one, and the main, it takes more than $2 billion US expenditure to discover a drug. Some of the drugs, even after launching into the market, they have to drop again within a 1 year of time, because different patients may get other side effects. Then if they are frequent, it has to be dropped out. So it is a very tedious process and expensive process. Then what I'm going to tell here, how the synthesis plays a key role in this drug discovery process. We have seen that nature is beautiful. Nature is giving wonderful molecules for many diseases. Then why we need to synthesize them? Why we need to make them in the laboratory? We can get it from nature, right? But it's not the case. As I mentioned, many anti-cancer drugs, if you take that halicandrin, when it was isolated from a sponge. A natural product scientist collected that material diving into the sea in a particular time of the year and get some 10 kilograms of material. After several different processes, he might have got maximum few milligrams of compound. But this few milligrams of compound is not sufficient to do your all required studies of 10 to 15 years. You need grams of material, kilograms of material. If suppose it approves, now you require in hundreds of kilograms, sometimes metric tons. Some of the drugs today, paracetamol, we need in metric tons. So you cannot get this many metric tons from the nature, right? So you need to have a method in the laboratory to make this compound from the available chemicals. So that is called synthesis. That is why we need to synthesize molecules. So what the definition is, if you are constructing a molecule intentionally by using the chemical reactions, that is called chemical synthesis. So if you are making a molecule, a total molecule, that is called total synthesis. If the molecule is from nature, you say natural product total synthesis. So there are a few other reasons why we are making natural products. One is the structures, the complex structures will inspire the chemists like me to develop new reactions. Because once you see the structure, you automatically start thinking how to make that molecule. And for students, some of the reactions like oxidation, reduction, alkylation, acylation. So these are the reactions we use in the laboratory. What you are learning today, you have to practice in future. But sometimes these reactions or the available conditions may not be suitable for a particular molecule. Then you need to develop a new method, new reaction. That is limited quantities of material from nature, as I mentioned. So you need to develop, have more quantities. Then other point is structure and stereochemistry confirmation. The natural product scientist will isolate the compound and he proposed one structure from all the spectroscopic analysis methods. But again, he is also a human being. He may also do some mistakes. Our mission may give sometimes wrong information. So keeping all these points, once you look at the structure and synthesize the same structure of molecule in the laboratory, then you can compare with the natural product. Sometimes it may come wrong and then you need to revise the structure or you need to revise the stereochemistry. So these are some of the reasons why we are synthesizing the natural products in the laboratory. So this is the best example one can show how the synthesis history began. And all of you know that urea we use in all our crops and mostly paddy, and you need metric tons, right? Thousands of metric tons required. But you see, when it was discovered urea in 1727 and it was isolated from urine. But is it possible today to have the required quantities of urea to isolate from urea? No way. You need to have a method. So it took 100 years after isolation of urea to make the molecule in the laboratory. In 1828, Frederick Wöhler synthesized the urea from available chemicals, silver isocyanate and ammonium chloride. If you react, you will get urea and you can make in the laboratory. This is the beginning of synthesis history. Then you know camphor. It is again isolated from alpha pinene trees. And today how much camphor we consume? In our temples and in many other places, if you see, again you need tons camphor. Can you cut that many trees and isolate this camphor from the trees? No. So again, Compass industrial synthesis, he developed a method in 1903 from, it's called semi-synthesis. Plenty available natural product you take and convert into camphor. That is again important milestone in synthetic chemistry. Then little more complex quinine. This is a first effective drug for the treatment of malaria. It was again isolated from plants. Then Woodward synthesized in the laboratory. He got a Nobel Prize. And then now today this was available in the required quantities for human health care. Then little bit further, if you come to 20th century, Professor E.J. Corey from Harvard University is again awarded a Nobel Prize in 1990 for the development of theory and methodology of organic synthesis. You can say that he's developed lots of reagents, lot of molecules he made in the laboratory. These are the best example here I have shown. You see how complex this molecule. It is not easy to make this molecule. Sometimes synthesis of these molecules will take, to develop a process for these things will take years together. So he was in the recent era in the 20th century. Then you see the structure of this molecule. This is the challenge for organic synthesis chemists today to make this molecule. This was again isolated from a marine natural source. You see how many rings are available. You cannot even number using available alphabets of 26 alphabets are not sufficient. You have to go back A dash, B dash, C dash like that. So today the scientists are attempting to make this compound in the laboratory. So from the history, you have seen how small molecules to complex molecule we are able to make in the laboratory. Because of development of science, development of novel chemical reactions, and development of technologies for their characterization, so many reasons. So then what, this is all general introduction, how drug discovery process needs time and money, what are the steps involved, then what is the role of synthesis, why you have to make molecules, and how the evolution of synthesis. Now, little bit I would like to show what we are doing at IICT. So my research group mainly involves in the synthesis of bioactive natural products. Then we also develop new chemical reactions, heterocycles and carbocycles. And mainly our institute mission and objective is to work with industries toward drug discovery programs, new chemical entities, or process development for high value APIs. So these are the three sub-compounds we mainly work to develop new heterocycles. I'm not going into the details. So these are the slides, 3 slides I'm going to show here. These natural products we have accomplished in our laboratory at IICT Hyderabad. These are all our natural products, tetrahydropyrenyl chemistry structure. And these natural products we have made in the laboratory with available chemicals. And you can see from 2008 onwards, we published in the different journals. So these are the molecules made by our PhD students. I choose the target and then decide the chemistry, and they struggle for sometimes 5 years, sometimes 3 years, sometimes within 6 months. Depends how we plan and how it is going to work. So, but in the process, they will get training by doing these molecules, how one should make a molecule in the laboratory, how one should plan. So then, little more complex natural products we made in the laboratory. These are all macrolides with various several asymmetric centers. You can see here one cladoscolide molecules we made in the laboratory. Then this center you can see cematopolide A and cematopolide B. This is one of the important. We found that the stereochemistry was projected in mistakenly by the natural product scientists, but after we synthesized in the laboratory, we corrected the stereochemistry. So these are all some of the examples, the natural product synthesis, if you do, what you can do. And then some of the alkaloids we have made. Mainly today we are focusing on this one of the molecule myrmecorin, and these are all achieved from a common available material, aspartic acid. In the flask you can see in between this compound we made for the first time from aspartic acid, and using this starting material we made all these alkaloids. So this is what epipatidin isolated from the skin of a frog. We again made this compound in the laboratory. This is longan lactone. This is again from seeds. I will show in coming slides. So I want to give just few examples to understand when it was isolated, how much quantity we get and how we can make in more quantities in the laboratory. So this is one of the compound cladoscolide D isolated from the fungus developed on the corn seeds. From this fungus, natural product scientists isolated this compound in 2001. So the challenge in this compound is this red color bonds, carbon-carbon double bond and this carbonyl group in this fashion is the key challenge here. So then I'm not going to describe any chemistry, but with available material in the lab, this is available in kilogram scale. So from this, by doing various reactions, finally we made this compound in 500 milligrams in 2014. You see, when natural product scientists were isolated, it was only 5 milligrams from more than a kilogram material. But where we can make in the lab required quantities, and if we required for further biological studies, we can make any quantity of this compound. This is what another compound, longan lactone. You see, 10.5 kilograms of seeds the natural product scientists collected. These seeds, in China, they used to use in traditional medicine for treating various diseases. It has a lot of anti-rich oxidant compounds. So this is one of the compounds the scientists isolated. You see only 3 milligrams of material. But there is no biological data available for this because lack of compound. So then you see in laboratory, starting from L-aspartic acid, in 5 to 6 different different reactions, we made 200 milligrams by taking 375 milligrams of L-aspartic acid. So if you take 1 gram material, easily you can make 600 milligrams. Like that, you can make whatever quantity you want. And all the stereochemistry, everything should appropriately match for this compound when you compare with the natural product. So by doing this chemistry, whenever we feel some problems or we need to develop in large quantities, large libraries of molecules, again, we develop a lot of methodologies. I'm not going to explain anything in this slide, but we make in the laboratory a lot of heterocycles from a common synthetic material available in the lab, this propargylic alcohol, and we made various heterocycles and published all these papers from 2008 until today. We are still working on this synthan, and this is another compound we developed in our laboratory, and now from this one single compound we can make all these varieties of complex structures, pyrocyclic molecules, heterocycles, and aromatic compound like this, and all these we have published in different journals. So this is mainly little bit useful for the faculty and also some student. What are the recent advancements in the synthesis? How the synthesis in olden days you used to work in the laboratory and you generate lot of waste material, byproducts, available catalyst. But what is today's trend? What is the today development after development of 100 years of our organic chemistry, organic synthesis? Today, the chemists are working to make the reactions in one part without isolating any intermediate so that you don't generate much waste. So these three things are playing major role: biocatalysis, organocatalysis, and metal catalysis. And then CH functionalization is another active area, because earlier if you want to functionalize a compound, you need to have bromine, chlorine, or any halogen example. So when you do the reaction, you will get a byproduct of HBr, HCl like that. But if you activate directly carbon hydrogen bond, you will not generate any such kind of byproduct. So this is another recent advancement. And very recently, this visible light and photoredox catalysis is playing a key role. Lots of publications every day are coming up. Even our research group started working on electrochemistry, so that you no need to use of any oxidants. Then you don't generate any byproduct. You just use anode and cathode platinum carbon electrodes and treat your compounds, and then you can do various functional transformations. So these are the recent advancements. So now you have learned how the drug discovery happens, and then how synthesis play key role, and what are the recent advancement and what we are doing at IICT. Now coming to drugs repurposing. Present pandemic situations, for the treatment of COVID-19, the best possible approach to get quick medicine is the only way, that is called repurposing. Repurposing means it has already existed, the drug has been already studied for safety, toxicity, all those things. So you no need to study all those studies. It is used for some other treatment. Now you have to see whether this can be repurposed to present coronavirus and COVID-19 treatment. So this was one of the approach chosen, and because lots of drugs are available for thousands of diseases, millions of people are taking, that means they are safe, and then institutes and companies address only some of the diseases. But now we have also seen that you need more than $2 billion 10 to 15 years time. So repurposing will take only less money. In short time, we can give the drug at least under emergency purposes. So studying the drugs are already approved to treat one disease, are conditioned to see if they are safe and effective for other diseases. So here for COVID-19 treatment, sorry, just a second. So there are two steps mainly involved under repurposing. You no need to study from the beginning. You need to screen the approved or marketed drugs for this present coronavirus, against coronavirus, and then you need to do little bit clinical trials for respective indication. So if you fulfill those two steps, it can enter into the market. So that is why it requires less money and very short time. So this is where again I have explained clearly about organic synthesis. But again, what is process chemistry? It is also synthesis, but in the laboratory we make only 100 grams or kilogram of material maximum. But when you want to convert into process chemistry, particularly industrial scale, as I mentioned, for paracetamol, we need metric tons. You know, each paracetamol tablet is 500 to 650 milligrams. And daily we need to take 3 to 4 tablets. So like this whole world, how much quantity it requires. So you need to make in a metric tons. That is where this process chemistry plays a key role. And these are the steps involved in process chemistry. Because synthesis in the laboratory is different. But when it comes to commercial scale, on a ton scale, you need to think a lot of things. The reaction should be very easy, very low cost materials we have to use, should not produce lot of waste, because industrial pollution in today's scenario, no way, we cannot accept anymore. And lot of other validations, so many things are required. Then you can make few kilograms to ton scale. So this is called process chemistry. And here this slide shows how this is how in the laboratory initially the reaction can be optimized. This is from our own laboratory. One of my students, Mr. Amul Patil, is monitoring one of the reaction. These are the recent reaction which we have developed for favipiravir, which I will just explain in after a couple of slides. And here again, this second picture shows the purification of the compound by chromatography. Then here again, you see this evaporation of the solvent using this rotary evaporator. Engineers involved, and they will set up the proper conditions, and they can do whatever we made in kilogram, here they can do in 1000 kilogram scale or ton scale. So then coming to IICT contributions, what it has 75 years of history, developing technologies for various drugs. Mainly once drug come into the market, if you have a cheaper and cost effective process, the cost will go down drastically. So these are some of the technologies we developed for various drugs. And you can see one of the important achievement when this AIDS virus came, AZT was the drug used for the treatment. Then the technology was developed at IICT and it was licensed to Cipla, the leader in antiviral compounds, and they have marketed all over Africa and other countries. And that was one of the milestone from CSR IICT. And another drug, this is called misoprostol, and this again wins the technology award in 2014, and this we have licensed to Avra Chemical Laboratories, and they are making and selling this product. So like this, lot of technologies have been developed by IICT, and what we have done recently in the last 6 months towards COVID-19. So as I just showed previously, repurposing. In January, February itself, we have studied lots of literature, and WHO also announced these are some of the molecules might be useful for the treatment of COVID-19. Some of them are already in the market, some of them are under clinical trials, some of them are available in only 1 or 2 countries. So, but based on the literature and earlier their properties, we felt that these are the 20 molecules we have selected. Under this, you can see we have categorized in 2 ways. One is available with already Indian manufacturers. This right side 12 molecules are already available in the market. If you want to use them for COVID-19, if they are going to work after clinical trials, the material is readily available. Many companies are already making. But the left side 8 molecule, if you see, in this 4 or 5 molecules are not in the market. That means no company is making. Particularly in India, they are not at all available. So, in case if they got approval for the treatment, we need to have a ready technology or ready process for their synthesis in large quantities. With that idea, we have begun our work in the thing is we cannot go out, we cannot move properly, we cannot buy the chemicals. Under those situations, we at IICT decided to stay inside the campus and somehow make available, process available for these molecules, and IICT worked on these 3 molecules in red color, remdesivir, favipiravir and umifenavir. None of these molecule compound is available in India, even for testing. If approved, the process needs to be, industry has to make process. So in this direction, these are the 5 molecule structures, and these 3 we have taken at IICT, and Mahender already mentioned that EIDD molecule by MRI University and this galidesivir, these are 2 under present clinical trials. So just to show this favipiravir history, how people thought that this might be useful for COVID-19 treatment. It was actually developed by Fusifilm. All of you know that Fusifilm we used to use in olden days cameras, the film. The Fusifilm company has a chemical company, and they developed this drug under the name of Avigan. It was approved since 2014. It is used only in Japan for the treatment of normal flu. If you get a cold and all those in normal flu, they are using as an antiviral agent, but it is only available and approved in Japan, not even approved by FDA. Then during this time in February 2020, they found that in China they studied that this might be useful for COVID-19. So in March we have immediately started developed process at IICT, so that if in case it approves, we can support Indian pharmaceutical companies with our process, so that they no need to spend again time. And we started in March 23rd exact date. I am the main project leader for this project. I'm happy to tell you. And then within 20 days of time, with the help of the students, which I showed a couple of slides back, Amul, Subbarao and those 4 or 5 people, we kept them in our campus. We did not allow them to go out because of this situation, and we provided food, and 3 shifts we worked. Within 20 days of time, we developed a new process with the available chemicals. Even if you want a new chemical, you cannot buy. You know, we depend most of the times for all raw materials from China, but everything is stopped. So with available chemicals, we developed a process, and on April 15, we transferred our technology to Cipla, and Cipla made the tablets and approached DCGI for their approval in May 2020. And then later, after initial clinical trials within a month, in June, initial tablet was approved for marketing by Glenmark in India. But you see, at that time, the tablet price was 103 rupees per tablet. Favipiravir, each tablet is 200 milligrams. So the dosage you need to take is huge amount of dosage. But with our technology,
Your
when
voice
we...
is not audible.
It is not mute. I don't know then. Now
No,
also
it is very clear.
okay. So when we made this compound and Cipla launched with our technology for 68 rupees per tablet, and now it came much down. But you see, we are happy that this is the photograph you can see here. I am transferring the technology to Cipla representative, and then even on the tablet box now here, Cipla clearly acknowledged our work. It is co-developed with IICT, and IICT logo is on this marketing box. So this is a great achievement by our group and our institute. Within 1 month of time, we developed the process and transferred, which is very efficient and cost effective. And now we transferred this technology more than 4 to 5 companies in India. And after making within the competition, the price became now almost 39 rupees per tablet within the span of 3 months. So this is one of the achievement. And the other molecule, remdesivir, which is again used for the patients mainly on the ventilator, advanced stage treatment. The injection cost is again very expensive, but this was all again a not approved drug before this pandemic situation. It was only developed by Gilead earlier for Ebola treatment, and in 2019 for 2009, and then they are working presently on a hepatitis C program. But during the situation when they got tested, they found that this might be useful for COVID-19 treatment, and initial results gave very positive. And then our Indian 6 companies got license from Gilead. Fine, Gilead gave license, but to make this compound available into the market, you need to have compound, right? So Gilead is not supplying them. Gilead is only gave a license. You can use my technology, you can market it, but you have to make on your own. Again, CSIR IICT started work on this molecule in the March itself, and by the time Gilead gave permission to these companies in May, we had this process ready with us, and we again happy to say that we are transferred to 3 to 4 companies, including Mylan, Cipla, and Laurus. So these companies are making this compound, and presently it is available in the market and used under the guidance of doctors at emergency conditions, mainly for patients on ventilator. So this is another compound we made, umifenovir. This was again an anti-viral agent and used only in Russia and China, not available in India. So we started this compound synthesis and transferred the technology. Presently, this is still under clinical trials in combination with various other medication. So approvals yet to come. Presently, phase 3 clinical trials are going. So these are the 3 important compounds IICT worked in the last 6 months, and 2 of them are in the market with our technology. And further, we are also presently still working under the leadership of our Director General, CSIR Director General, Dr. Shekhar Mande, and our Institute Director, Dr. Chandrasekhar. 2 dynamic leaders have launched this COVID-19 mission for the synthesis of different APIs. So you can see our CSIR has 38 laboratories across the country, and these are the 9 laboratories works under chemistry specialization, but the main labs are IICT and NCL. These are the 2 main laboratories in the country for working this process demonstration and technologies. So these are the around 13 molecules still we are working for their cost-effective synthesis. So in addition, IICT has also played a key role under this pandemic situation from March by making sanitizers, by making masks, and distributing to mainly police and GHMC, municipal market yards, where they are working under this pandemic situation. Without their work, we could not be safe and clean today. So we all supported them with our sanitizers and masks. So these are some of the pictures. So with this, I hope I be able to demonstrate that mainly for the students, how the drug discovery takes the time, how the drugs will be discovered, and what is the role of synthesis, and how IICT is developing new synthesis of molecules, and further we are converting them into process development, and what are our recent contributions to COVID-19. So these are the things. Still various other departments are there in IICT working under different specializations. So with this, I must acknowledge all my research group members since 2008 working under my supervision. Only I can give ideas and thoughts, but they are the one have to go and work in the laboratory day and night. So far 18 students have been graduated, and most of them are working abroad for their postdoctoral studies, and presently 12 members are pursuing their PhD under my supervision. So for this, always all the directors are supporting. So since my join in at IICT from Dr. Yadav, then Dr. Lakshmikantam, and presently Dr. Chandrasekhar, these are the directors, supported me, and mainly financial support for our research is from CSIR, our parent organization, and we also get funds from Department of Science and Technology, Ministry of Earth Sciences for drug discovery programs. So with this, I conclude my lecture, but before that I wanted to show some of the pictures mainly for undergraduate students who mainly don't know about the IICT, how the IICT history. So it was actually established in 1944 by Nizam's kings of Hyderabad before our independence. You see, the Nizam king thought that such kind of laboratory is required to support industries those days. And then after independence, the foundation stone laid in 1949. Then the first building was inaugurated in 54. Then it was renamed as a Regional Research Laboratory, RRL, in 1956. So later, because we are developing a lot of technologies, then it was renamed in 1989 as Indian Institute of Chemical Technology. So to reflect our national character. And then these are some of the pictures. You can see Pandit Nehru while inaugurating the building, he is addressing all the staff. So after that, IICT has grown up with lot of specialized buildings and specialized centers, chemical biology, discovery laboratory. You can see the natural product lab, pheromones for agriculture, agrochemicals and fluoroorganics. Like this, you can see plenty of things, and with all the world-class analytical facilities we have at IICT for characterization and all the technical development, all the available instruments with latest developments. These are some of the instruments, pictures I am showing. And then this is one of the unique facility in the country we have at IICT. It's called National Mole Bank. So in this bank, we don't keep money. It is a mole bank, molecules bank. Whatever molecules we are making, we keep in this bank, so that this is a property. Molecule is also a property. Today it may not be useful, tomorrow it may be useful for another disease. So with that intention, we are storing all our molecules in this bank under safe conditions at -80 degrees centigrade. So inside the mission, it will be like this. Everything is robot controlled automatically. We give sample, each sample with a barcode, and when we want to retrieve, we can retrieve with barcode, and then it will be tested for various diseases when the new target identified. So this is how the screening works. I'm not going into details. So finally, I would like to give one message to my dear students. There are no shortcuts, only hard work. You have to do and train hard and be patient. It will pay off. Now the choice is yours. If you go for easy work, you always complain, you always blame the system, you tell lies and you cheat the people and you pretending to do work. But hard work is, you have to get inspired by these things, and you have to mentor your younger people, and then you help the people. always learn. Today also, I have to learn many things. I have to learn what happening around the world. I have to learn. Otherwise, I can't perform or pursue my research. So, and you need to learn the giving nature, and then this is the hard work uh, uh, characters. So now the choice is yours. Never underestimate yourself that I am from this background, I am from that background, I am from Telugu medium. Nothing stops you if you want to learn, if you want to get a success in your life. This is the only mantra for success: hard work. That pays you, maybe sometimes little early, sometimes little later, but hard work always pays. This is my message to all the students today. Thank you one and all. Please stay safe and keep healthy. These are the 5 rules we should follow under this present pandemic condition. I once again thank Telangana Social Welfare Residential Educational Institute Society for giving me this opportunity, and this is a wonderful opportunity. I always love to teach students. Even during my graduation studies of BSc in Karimnagar, I used to do tuitions for 9th and 10th class students at Telangana social welfare hostels, residential hostels. Morning and evening I used to go for science tuitions. So again, it's reminded me those days. Thank you one and all.
Thank you, sir. It's a beautiful lecture, sir. We had an overview of the complete chemistry, I believe, starting from the importance of chemistry to how at present chemistry is used by each and every person. Thank you very much, sir. And you were touching about natural products. I have done research in natural products, so I was feeling very happy to listen to those words, and then flavones and coumarins, all these things we have done it. Chalcones we have isolated, flavonoids we have isolated. It is very nice to hear from you, sir. And now we could know about IICT very clearly. Such an advanced lab, we have it in Hyderabad by the side of our institutions, and still we are not utilizing your facilities, I believe. Therefore, we request you all, let us have a collaborative work, and then you please use our human resource. So many children are here. You can use our children for small projects and all, and together we will achieve great goals, sir. All whatever is possible, we'll sit once, and then I think it is high time together.
Most welcome for this. I'm happy to do that, and every year we have, throughout the year, we are opening for the graduate students. And once the situation becomes normal, your college students are most welcome to visit our institute and get the flavor of all the facilities, so that at least a few of them will inspired towards science and research activities. And I am also giving a lot of inspiring lectures, various schools and colleges. So you are most welcome, madam.
Thank you very much, sir. Now the session is open for question and answers. Is there anyone? You please ask if you have any doubts. Session is open for question and answers.
Dr. Raji.
Sir.
Hello.
Yeah, sir.
Am I audible?
Yeah. Please go ahead.
Please go.
Thank you very much, Dr. Raji Reddy, for your excellent lecture.
Yeah.
Very useful, highly informative, and I am very much confident that all our young students definitely followed each and every point you said in your lecture, and it is a very lucid manner you have explained. Thank you very much. And in 20 days time, you have developed a process for congratulations to you, and congratulations to your director and the entire team of IICT. And thank you very much for accepting our invitation and delivering excellent lecture for the benefit of many students of social welfare degree colleges. Thank you very much.
Thank you, sir. Thank you.
Yes, sir. Now that you have synthesized these medicines, can we get these COVID-19 medicine for cheaper?
Yes, yes, madam. It is
Maybe?
already, yeah, it is already available in the hospitals. But only thing that, especially remdesivir, is given permission under emergency conditions only, because that this was not earlier in the market. But since it is working presently for this COVID-19 treatment, so the patients are getting this medicine, but under the presence of doctors, it is given at the hospitals.
Yeah, yes, yes, sir. Great job, sir. Great job you are doing.
Sir,
Thank you.
Yeah,
sir, this is Dr. Shruti, student of Dr. G. Sudhakar, sir, from IICT.
yeah.
Sir, you said that three drugs we are only synthesizing in IICT. Nowhere in India, no other companies synthesizing.
Now, before they approved in the market, it was not available in the Indian market, or Indian companies are making these three drugs.
Okay.
Now, once it is approved, only companies have to synthesize and develop, right? It has to be synthesized under approved manufacturing facility. IICT cannot synthesize in the lab and market, right? It has to be a safety environment place. But we have developed the process. We have made the first sample in India.
Okay, sir.
Yeah.
Thank you.
This is Mahender. I have a couple of questions to comments and questions to my friend. My friend Raji Reddy did an excellent job. This is called bench to bedside. What he showed is what's happening in the lab, how it is transformed in the company industry mode. This is an excellent job. And also he has given very excellent overview about how chemistry influences the entire world, or our regular routine day life. In fact, in biology also, when you talk about biology, that relies on chemistry as well. See, let's say DNA, RNA, nucleic acid. When you say nucleic acid, immediately a chemist think about a sugar, a base, a phosphate, right? So Raji Reddy, congratulations, you have done a great job. I'm very happy that my friend is doing excellent. One question is, so how IICT is helping the undergraduates to grow as a matured scientist, or influencing them to come into the research side, or become a scientific world? I would like to hear your comment on that.
Yeah, thank you, Mahender, for your wishes and your compliments. We are encouraging students to take summer internships mainly, whether in that their curriculum it is there, or now most of the curriculums have these internships. Even though if they don't have it in their curriculum, but during the summer vacation we are offering some brilliant students and interested students. Every year we are selecting from each college, and they can get the real experience in the lab by working for month or 2 months, and then slowly, based on their if they get interest and motivation, obviously many students are writing back to us, or last year I worked in so and so lab, I want to learn more or continue. Like this we are getting things, and that's how we are encouraging these undergraduate students.
Excellent, amazing. I think the social welfare residential educational institutions societies should take this opportunity, utilize the opportunity to introduce their students into this kind of environment. And also, Raji Reddy, one quick question. Yes, undergrads you guys are doing. Are you guys thinking about any high school students, like an intermediate, or any others, like a quick introduction to the lab rather than in-depth research?
Yeah, so we are in that direction. Earlier we used to have only open day during our foundation day, but now in the recent times, last 5-6 years, our director opened throughout the year. Every day, few schools and few colleges are coming to the institutes and looking at the facilities, and couple of our dedicated staff are addressing them, how we are working and how you want to, if you want to come, what are the opportunities. So with these explanations, we are doing this activity for school children and intermediate students also. And we ourselves going into the colleges and schools. Recently, before lockdown, even I visited one of the Jilla Parishad High School in Karimnagar district, and we did some activity. This is actually part of our student curriculum, their PhD curriculum. They have to do some kind of social responsibility activities. For that, we are giving 4 credits in their course. So that is not a study. They have to visit some schools and other things and show our demonstrations lively, how we are purifying compounds, how we develop. So to just inculcate some kind of, in their mind. So this is part of our PhD curriculum in CSIR.
Excellent job, Raji Reddy. Wonderful, excellent. Yeah, it's 3 a.m. now. I waited to hear your talk, and you did an excellent job. I wish all the best to all the students from residential colleges. You guys have done a great job. Thank you.
Yeah.
Thank you. Thank you, sir. Yeah. I think Raji Reddy garu, what I was feeling is you are doing a very good research. In somehow we want to collaborate with you. I want you to utilize our students also, because for your studies and all, you can utilize our students. They can get the data so that you can compile, and then extensive research can be there, and that sampling can be a large sample you can collect, I believe. Can that be possible? You please think over it so that we can involve them, not only looking at your labs and all. We can involve in your process.
Sure,
So,
madam. We will make some MOU with this college, and then we can
we can create interest in pure sciences. Now people are going away from pure sciences. So it's all applied sciences, and then computer courses and all. So let us together work for that one, because India needs a lot of good brains now. So we should stop brain drain also. And I hope you will take the initiation and then do the needful at the present for the present society to grow. Thank you very much, sir.
Certainly we will take up.
Rajni Garu, just one comment here.
Yes.
This is Mahender again.
Yes.
So a couple of friends of mine, and we came here, and every day we are discussing about, opening opportunities, overseas opportunities for our students. So there's a lot of discussion is going on. I will keep you posted, and I would definitely prefer the residential college students. Certainly.
Thank you, sir. Definitely will help you. And we are also on the same job, sir. We are looking for such things.
Yes.
Recently, our JS sir and then Secretary Garu has spoken to the government, and they have government has allotted 100 overseas seats to our students, social students, for the through government. Usually 300 students will be going overseas. Out of 300, 100 are from today on, from this year onwards. They are completely given to the social welfare. Preference will be given to social welfare students.
Excellent. Do you have a PG programs as well?
Yeah. This year we have introduced chemistry. Also, we are introducing this year.
Okay.
Chemistry, mathematics, statistics. 5 groups we have introduced, sir.
Biotechnology.
Yeah, biotechnology. Slowly, we want to grow the social welfare into a university, women university like. So that is the vision of our secretary, sir. But we have missed it today because of our mistake, little bit of mistake. He was very much interested, but really I felt sorry. He is a very fine person. He has transformed the education, sir. Our Indian education into a world-class education with the limited facilities, whatever we have. So we are very happy to have him. Thank you very much. I really thank both of you. And I'm happy that you are from OU. I'm also from OU. You are also from OU. And then from our area, Telangana, you are from.
Yes.
So many common things are there. And we are happy to see you all in that position. And we'll be more happy if you interact with our students and make a transform for students.
Anytime. Just schedule some time. I'm happy to do so.
Yeah.
Always.
Definitely, sir. We will invite you for scientific committee we are planning to have. Really, we want your presence also in those committees. We have gone away from research a little bit towards academics. Therefore, we want research hands like you people. and we'll be very happy, sir. And I thank from bottom of my heart, really saying so. But two of you are marvelous, and then beautiful lecture we had, very nice, down to the degree college level. Clearly you have and lucidly you have explained. We are very fortunate to have, and I should thank Professor Ashok for this. Professor Ashok is my classmate and lab mate. Therefore, he has come forward.
Thank you, Dr. Reddy.
Yes, and then special thanks to you, Ashok. Really, we are grateful to you. And
It is my pleasure.
I will not leave Ashok now. Still, I want to take him
No,
along with
in fact, you have given an opportunity to me. In fact, you have given an opportunity to me, and both of my students made this webinar a grand success by delivering excellent lectures. Hearty congratulations to Dr. Mahender, Dewalji, and Dr. Raji Reddy. Thank you.
Thank you very much, sir. Now I request Sirisha to present memento and give vote of thanks to Raji Reddy garu. Sirisha?
Yes, ma'am. Yes, ma'am. Thank you, ma'am. A warm greeting to one and all. It's my pleasure to present vote of thanks to our honorable guest, Dr. Chada Raji Reddy Garu, for sparing his valuable time to be with us. Sir, I want to express my heartfelt gratitude for enlightening us how we are connected to chemistry in our day-to-day life. Thank you for showing us how the natural products were used since ancient time and how this is being processed in the laboratory and the recent technologies used during this COVID-19. It's a very informative session, sir. Thank you for bringing science to the doorstep to our students. And I strongly believe that your team at IICT will surely bring potential vaccine for COVID pandemic. I think our student community, teachers, thinkers, and common people all around came to know how our system is working on combating this pandemic. Finally, your presentation on IICT enlightened our student community and also motivated them to aspire to become a scientist like you, sir. Thank you once again.
Thank you, Sirisha. Thank you very much. Now, I call upon Dr. G. Nirupa, Principal, TSWRDC, Mahendrahills, to present vote of thanks.
Thank you, ma'am. Thank you for giving me this opportunity. Right. This COVID-19 outbreak has affected all of us in one way or the other. It has disrupted the way the institutions, the government, and the companies all across the world operate. So this is a time when we require a proactive and flexible and innovative approach. So here the role of chemistry is vital, and in the course of time, the increased production of disinfectants, diagnostic tests, ventilators, protective masks and gloves, gowns, everything, they have been increased. Their production has to be increased. So all this, why I'm trying to say is we have to understand the crucial role the chemists play, and more than how the chemists are more than willing to provide solutions in times of crisis. So hence we planned all this platform to enrich our young minds with possible chances to merge knowledge of chemistry in finding possible chances or to merge in the field of in finding possible potential vaccine or something like for corona. So it was a very wonderful experience and all by listening to all of your lectures and all. So both Mahender sir and also Raji Reddy sir both have done an excellent and a wonderful job. And the way that you have explained, Mahender sir explained the types of coronaviruses and how they have been transmitted, on different types of coronavirus, how it is transmitted, and the different genomic organization of coronavirus, and the mechanisms of virus 2 type of immune systems, everything it was. And also you have given enlightenment of how the clinical trials of vaccine in different countries have been done. So it was a very, very important, and then it was a very, very useful, knowledgeable session to all of us. In the same way, also thank Raji Reddy Garu also the way he spoke about the different types of drugs used, and we normally now we are being heard that the immunity boosters and all to be done there, and how the Ayurvedic this one Ayurvedic medicines and all can work for them also. So this kind of things also can be done in the quite in the new research also, and the process. It was a great achievement in that is in by providing a synthetic process for favipiravir and remdesivir under the pandemic situation in a very short time. So it was by seeing all the equipment in your IICT lab and all, it was like brainstorming for all of us, and it was giving a boost for all of us to enter into this field. Like, so I hope it was quite inspiring for not only for us but also for the students, because it is the young minds who they have to think about the next and all. So this was a very, very knowledgeable session and quite inspiring way. It was a very wonderful session that I can say. So in this way, I thank all the two wonderful speakers for their eminent and tactful way of dealing with all the lectures and all. So thank you so much, sir. And I hope the session was very, very useful for everyone. And because it was a YouTube link, we can share it every time and just think about and see once or twice and then share it with many people also. So thank you so much, sir. And I take this opportunity to thank Ashok sir too, because he was the one who has like introduced all these the eminent personalities over here to and made the this platform webinar of grand success. So thank you so much, sir. And next, I also I thank Secretary sir. Though he couldn't come into the platform and physically he was not there here, but I think because of the inconvenience, I think all of us know that. And really, I really apologize everybody for that the technical issue where we had a uh, we had. And kept you all waiting for a longer time and could not join the eminent personalities like Secretary Sir Garu, Praveen Kumar Sir Garu, and Praveen Mamdala Garu. Anyway, they can make he came and joined. So I thank the Secretary Sir for his quite inspiring ways. The way he is a boost for all of us. The way we work, what we are thinking today, and what the way of work today. It is all inspiration because of that supreme Sveru, I can say. And Praveen Mamidala Garu also. He's another. He's also a great inspirator to all of us because of his able guidance. We are all the degree colleges and the higher education is being done. So I thank all the two dignitaries from the social welfare organizations. I also thank our Bhanu Prasad sir, who is OSD of our degree colleges, and for making convenient to come and join this program. And also, he has also been quite advisor. He's been a chief advisor to all of us. And thank you, sir. And I also thank zonal coordinator, ma'am, Dr. P. Rajini Garu. She has been an excellent inspirator for all of us. And because of her, I mean, she had been organizing all these webinars and seminars and all. So she's a quite inspiring personality, I can say. So thank you, ma'am. Thank you for everything. And the faculty and the staff of Mahendrahills, I also thank you all for making this program and all these technical issues and all whatever are there. So it was quite interesting way you have you have presented the things, presented the webinar. So it was a wonderful session from your side too. I also thank Vishnu. Vishnu has been guiding us with all these technical issues of creating all the what are animations you can say and all these things for making the webinar much more interesting and more presentable. So I thank Vishnu also. And I also thank the principals of other colleges because of whom we have got excellent and a wonderful registrations. And by now it is the registrations have reached 1600, sir. And because we did not expect so much and huge response from the students and the other outside people also. So because of that tagline of that tagline of our invitation. And also not only that, by seeing the speakers profile and all, there were huge line of people they were waiting to come and join. So because our Zoom thing was taking only 100 people, we could see only 100 people can name in that things. Though they were not participating them, we were able to see only 100 people. So by the time we were able to remove each one by one, we were removing, and we could not reach all the 1,600 students also. So that was a great major issue why we had the technical issue, because it's the first time so many people have entered into. And we here also we have learned so many things, and how to go forward for the next programs or next webinars which have to be conducted. That is also we have learned how to deal with all these things. And for that, I thank all the principals of other colleges also for making this program, or for making all this, for taking this into every student's view. And next, I would like to thank all the participants and the students, whoever have participated and given the valuable feedback and all. I thank all of them, and also for the students' parents also, who have been quite they have been supporting. They have been supportive to the students to join to the webinar, and providing them with the necessary gadgets, cell phones, and so for joining this webinar. So thank you each and everyone. If I have forgotten anybody, and just kindly excuse this. And I hope I have everybody. I'm thankful for everyone. For those who have forgotten also, please, I thank everybody who have been associated with this whole webinar session, and making this webinar a grand success. And thank each and everyone from the bottom of my heart for making this webinar a very grand success. Thank you, sir. And once again, Mahender Dewal Garu, I want to just mention the way you have been waiting. I mean, it is 3 o'clock in the morning, as you said. So at this week time, you are there here to inspire all of us. You are there here to help us out with all the things. And also, you further also with you have also said that you are going to help us out in the further overseas education or something, and also not only that, in research and all these things. So I thank you, and even Raji Reddy Garu for his positive attitude towards such degree colleges, and trying to collaborate or trying to initiate that MOUs and all. So it was very a wonderful session, and great dignitaries have a great heart that has been proven again by this session. Thank you so much. Thank you each and everyone, sir. Thank you so much. Thank you, ma'am.
Thank you, Dr. Rupa. Yeah, Ashok, Professor Ashok wants to speak.
Dr. Rajani.
Yeah.
Yes, one minute.
Yeah,
it is unique opportunity for me.
Yeah.
Convener of this webinar is none other than my own classmate, Dr. Rajani, and lectures delivered by my own students, Dr. Mahender and Dr. Raji Reddy Garu. And just now I received a certificate of participation of this webinar signed by my classmate, Dr. Rajani, and my own student, Dr. Nirupama. Thank you very much to all
Thank
of you.
Sir. I really thank you, sir. Because of you, like you had been on inspiring every time, the way you had been from long back, and now also you haven't changed in your temper. I can say you given the way you come down to all of us and talk. It is really inspiring. So again, you have proven that great dignitaries are always having a positive and a down to earth thing. Thank you, sir. Ashok sir is really wonderful having been associated with you. Thank you, sir.
It is a very rare opportunity. Nobody will be experiencing it except Professor Ashok.
Yeah, all the students and classmates and students in the webinar.
Yes. Thank you, Ashok. Special thanks to you. And I want to announce the participants. All the participants will be getting the certificates if they fill their feedback form. Now we'll be ending this international webinar with the national anthem.
Good afternoon, one and all. I request everyone to stand up for national anthem.
भारत भाग्य विधाता पंजाब सुंदर బలవంగా వీర్యం తవ
तव शुभ आशीष मांगे गाहे तव जय गाथा जन जन मंगल दायक जय हे मानस भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Jai Hind.
Thank you. Thank you.
Hearty congratulations to Dr. Rajani and Dr. Nirupama.
Thank you, sir. Thank you. It has been possible only with your support, sir. Thank you.
Ma'am, super program.
Thank you. Thank you. Thank you, everyone. Mahender Garu, good night for you. And then others, bye-bye. We'll meet next week in another webinar. Thank you.